Hey everybody, welcome to the Growing with Fishes podcast, episode 256. This week we have Mathew Gates back with us again, IPM expert extraordinaire. Thanks for joining us.
Thanks for having me. Thanks.
It should be like an official title.
Right? We gotta, you've been back enough times. We gotta give you like some kind of.
On Twitter, I thought it was clever to say Insectus Optimus Maximus, but that's a bit of a mouthful and a little pretentious.
It's great.
So I'll have to work on that.
It's great for Twitter. It should be a little pretentious for Twitter.
We also have Marty Waddell joining us tonight, AP Meds.
Hello.
And Fumidor.
How's it going? Cheers, guys.
We have a little bit of housekeeping real quick first. We have, see if it'll load right. No, it doesn't want to load right. 2 seconds. I'm sorry, guys. I fucked up.
You should do like on my show where we say, you're welcome. Basically, no apologies. If you guys are watching, screw you. Like, you're welcome.
Be sure to check out apmjclass.com if you're looking for a full top to bottom aquaponic cannabis class experience. Marty and I have added new content to that almost weekly. So definitely check it out. We have over 600 slides and counting. Actually, I think we're way past that now with a lot of the live sessions and everything else. So definitely check that out. Marty's got a bunch of new content with his new greenhouse build, and we've just finished recording a ton of new content for the channel. So definitely check that out. And then we also have, you can see in my background, the virtual Aquaponic Cannabis Conference, November 13th and 14th. We'll be announcing the speakers next week. I'm just confirming the last 1 or 2 to make sure their times work before we do the announcement so that I can give you the schedule as well. I think 3 people left, I think, just have to confirm their times. And then we'll have that out. But we have over 30 speakers for that conference and 26 hours of education. It's going to be 8 a.m. to 10 p.m. And just an absolute like wall of education. So check that out a little bit later on. So thanks a lot for joining us tonight, Mathew. You actually have a lot of really cool stuff you've been working on. You had a new paper or book coming out on viruses. Why don't you tell us a little bit about that and what you've been up to lately? And then I got a bunch of really awesome questions for you on viruses and a bunch of other fun stuff.
Yeah, absolutely. Thank you for having me. So to answer what you were saying, I have 2 chapters of for 2 different books. I have a colleague who's publishing books about organic agricultural practices. And one of them was about arthropod pests in cannabis, and the other one was about viruses, basically cannabis virome. And especially on the latter, but also in the former, I had to be pretty careful. I couldn't go over as much as I wanted to be. Normally they had a pretty tight limit. So I had to be very clever with my wording and kind of keep it brass tacks. So, and I was very excited to talk about this because I think it's something that's pretty underrepresented in the cannabis space for a lot of different reasons. But yeah, the more people I can kind of at least get even to the concept of thinking, oh, I might have a virus, and what could that actually be in this context, I think is very important. How that's vectored and that kind of a thing.
So I think a good place to start is, can you explain to people the difference between a virus and a viroid? Because people often use them interchangeably, but they aren't really the same thing.
That's true. So like they're all viral agents. And so when I talk about them in the plural, both of these kind of groups that are very similar, I sometimes use the word viral agent or something like this so that I can, be specific. But basically viroids, they don't have a really common thing that viruses have, which is a protein sheath called a capsid. And viroids are basically like naked in that way. And that's the kind of the main difference. Viruses, I think, as far as we understand it, are a lot more prevalent. And if I am remembering correctly, I think viroids are only found in plants so far. I don't think viroids infect other or colonize other kinds of organisms, but if they do, I mean, I'm not saying it's not possible, and I'm not a virologist, but if they do, I don't know of any examples where that's the case.
So what are some of the different common ways that cannabis viruses are vectored? I know I've seen pruners transfer them, at least with mosaic virus firsthand at facilities. What are some of the other different ways that and vectors that are, spreading this virus right now, or various viruses? Get to.
Right. So like in that chapter, I talked about a few different viruses, and I also talked about the hop latent viroid regarding cannabis. So typically viruses are actually, especially in plants, are vectored by insects. That's probably the most common route. And a lot of common pests are also vectors of viruses, and usually insects. In some cases, plants can be like, there's a rose rosette disease that is transmitted by a type of russet mite, not the hemp russet mite for cannabis, but a different one for roses. So there are various kinds of arthropods that can vector viruses, theoretically, conceptually. But for cannabis in particular, you want to consider the silverleaf whitefly, which is known to vector over like 480, I think it is, plant viruses. Super common in cannabis, although I don't see it as often as many other pests, and also many other plants for that matter. So the silverleaf whitefly, and it vectors the lettuce sclerosis virus for cannabis, which is also kind of a new discovery.
Go ahead.
Oh no, I was just gonna say, and the beet leafhopper vectors the beet curly top virus, and those are like kind of the two main big ones that people should be aware of. That's becoming more prevalent in cannabis.
I was going to say there was two different facilities where we suspected lettuce sclerosis virus in aquaponics, and we did not have at least any detected whiteflies. And we are wondering if there isn't some other aquatic vector or some other insect that maybe we were, unaware of that transferred, or maybe even through just people being stupid with pruners. I could totally see that being the case as well, not paying attention or not following procedure.
Yeah, definitely right. Our understanding of the pathology is going to be sort of unsupported for now until we get more research. In this case, the LCV was discovered in Israel, and I'm not sure exactly off the top of my head what the systems or the dynamics of that were, but I do have a video about it on my YouTube channel for people who are interested in that information, and I kind of lay it out more articulately. I even use my own videos kind of as notes sometimes, so that's something that people could do if they wanted to learn more.
Awesome. No, that's great. I love the content. In fact, if anyone hasn't had a chance to check it out, he has probably one of the coolest videos on the internet around pest management, where he goes over the evolutionary history of cannabis's ancestors and the pests that were attacking it at the time, according to the current understanding. And it's one of the most informative and most interesting talks I think I've ever heard on cannabis pest management. If you haven't checked out that YouTube video on his channel, please go check it out. I don't know if you want to name the name of that video because I can't remember the name of it, but
it's a long name because I hate marketing, I guess. But it's part of, I started as a series. It's the global cannabis pest management review, and that was the 2019 parts 1 and 2. So part 1 talks about the evolutionary history, and part 2 talks about pests and ecology, or ecology and pest host relationships. And it goes through from itty bitty viroids and viruses all the way up to arthropods and mites and insects and that kind of a thing, various microbes, along that path, so that people can kind of understand all of these various organizational scales of life that interact both positively and negatively. I feel like it's kind of it's sort of a pernicious and persistent perspective about ecology that like there's like and even I'm a victim of it when trying to describe things as like good bugs or bad bugs or good things or bad things. And like if you have a balanced environment, that means that you can't have bad stuff lurking kind of in the area. And I think that that's almost true, but not totally always the case. Like the context and mutualisms and parasitisms can change over time, especially with microbes. And there's a lot more, research being done every day about that kind of stuff. So I like to keep it holistic.
So what are some of the different viruses that people are finding in cannabis? I don't know if you want to maybe start with, we can touch on this. We actually talked about this last night on Fumi's show. The new dark horse data that 90% of California facilities have hop latent viroid. That's a lot of facilities.
Yes,
right? So,
it is. When I talk about cannabis viruses and also kind of pathogens in general, bacteria, fungi, and all of that, sometimes I'll get people who say things like, if the plant's like too healthy, or if plant's healthy enough, it won't have a problem. Or that like potentially it's a bit of a conspiratorial perspective that the nurseries and the clone and the tissue culturists and those folk are very keen to spread this information. How convenient that it's their job to remediate these plants. And they're also saying there's this massive need to do so and to check and follow the money. And I mean, I think that it's actually valid to have that sort of skepticism. I definitely I applaud that, to like not take something on face value. But I'm also inclined to agree with the results of like this article, for example, that at the very least, especially because hop latent viroid has such a very specific set of symptoms, or even kind of lack of symptoms, that makes it very difficult to track. And since prohibition has made that very difficult already to track things like pests and that sort of a thing, which is why I try to help as many people as I can, like by coming on the show and talking about it. I'm just not surprised. I guess I wouldn't be surprised at that level. 90% is very high though. So, I mean, the implication is it might be a little bit more than that, right? Because it's not a census.
So, what are some of the other viruses that are being found in cannabis? I know there's quite a few different mosaic viruses, and you've identified a whole bunch of others. Tell us about the different types that are being found in terms of varieties.
So there's the hop latent viroid that a lot of people have heard about recently, and it is a viroid, so it doesn't have this capsid protein shell, and it's very persistent. So the latent and hop latent viroid means that it has sort of a latency to it, which means that you don't always get the symptoms immediately on colonization with the host. And in some cases, the symptoms that you get are actually quite mild. So especially, unfortunately, for people who are particularly new, right, and maybe don't know what a really nice, healthy, vibrant plant might look like, or if they're likely to interpret their first run as not very good, they might mistake those symptoms like stunting and like short growth and spindly branches that might snap and this kind of a thing. These sort of generalized characteristics they might interpret as something else. So I think that's kind of what makes it one of the things that makes it really problematic. There's other viruses too, or other viral agents, like I said, beet curly-top virus is vectored by the beet leafhopper, and that's very common globally. It's in North America in many places, California, even in the Midwest. I have a video that talks about the beet curly-top virus's presence in Colorado and also in Arizona, for example. And I've also had people tell me that in California they've encountered it as well, which is sort of unsurprising. And then there's the sclerosis virus I already mentioned. That's vectored by the Bemisia tabasi, the silverleaf whitefly, and that's a crinovirus. Those are mostly associated for whatever reason with kind of southwestern North America, but this was found in Israel, so obviously very, very far away. But a lot of horticultural trade is likely to spread these pathogens and their vectors around quite a bit. There's also a virus that actually people don't have to deal with too much, and that's Cannabis cryptic virus, which I think is kind of an interesting virus to talk about. I might have talked about it before as well, but essentially,
So beak curly top.
yes. So here's some very mild symptoms of that sort of leaf curling. And as it gets more and more pronounced, you'll see that the leaflets will kind of curl on each other like very severely. So the Cannabis cryptic virus is interesting because it's not actually vectored by anything. It's it stays in the plant and it gets vertically translated or transmitted through progeny. And it's interesting because it's very closely related to some viruses associated not with plants, but with fungi. And one of the sort of speculations is that perhaps it got into cannabis a long time ago, originally from a fungal pathogen. And then when that fungal pathogen colonized the plant, it moved into the plant this way, which happens to other things like genes and sort of stuff like that. So it's not outside the realm of possibility. Pathogens sometimes just kind of get transmitted this way. But because that's not how it normally transmits, it kind of got stuck and it's just kind of there. I guess it's associated with certain hemp lines, which are becoming more and more popular in cannabis breeding. So I expect to see it more often, but who knows what negative effects it really has, if any.
Well, they think that THC it was originally, like way back. I was reading about or maybe Steve was talking about it. I don't remember that it would have been a response to a viral infection that originally.
Yeah, they traced the THC gene back to a virus, if memory serves me correctly.
I think that's the case. I also remember reading about that as well. I like to point out that some of the ARC genes, ARC, that are related to short-term memory in humans is part of I think a retroviral interaction. So we have a lot of, and also I think this is the case for the placenta, actually, mammalian placenta development is, I think, also thanks to a retroviral interaction. Pretty crazy stuff.
So is the gene that makes you a boy or a girl?
I actually don't know if that's true. I don't think so, though.
I remember reading that.
Oh, maybe so. I really don't know. I know that the Y chromosome has some weird stuff going on with it.
Yeah. Let's talk about expression. So this is something that I found really interesting observationally. I was at a grow facility that had the same lights set at different light spectrums and in a block with 4 different setups. And one of them was set to a different setting. And you could see the one cultivar just under the one light spectrum was expressing mosaic virus in just the one cut and under just that light and not in the rest of the room. And you could see how it had smaller buds, longer internodal spacing, smaller curled leaves, the patterning, the whole nine, just underneath that one light. Everything else was the same. They came from the same mom. I thought that was really interesting that the light was having that much of an effect on the viroid load and the viroid expression on the plant, like some kind of other stress trigger was going on with that bed, with those lights. I don't know. It was, again, strictly observational. I don't have anything beyond just being there to see the place. But I thought that was super weird, but also I thought it was an interesting point to bring up how viral expression can be dramatically different from even one part of the plant to another part of the plant, and have, be real messed up over here, but not so much over here. And a healthy plant can oftentimes, suppress even an infection with certain viroids. Do you want to talk a little bit about that and how, like, especially when it gets to some of the mosaics and stuff like that, there's a lot of bad misinformation on, like, hey, well, it would be all over the whole plant and it would kill it right away. Well, not necessarily. It might be able to kind of reach some kind of balance. And there's all different types of mosaic viruses. There's Arabis, there's alfalfa, and all the other cucumber that you can test for now. And we'll talk about that in a minute with the different testing. Do you want to talk a little bit about how that dramatically affects what's going on?
Yeah, absolutely. And I have a research report in German that I shared on the Chief Home Grow podcast that people can check out there. But also, I'd be very happy to share it here for those who are interested. That they did experimentally expose cannabis to various viruses, and a couple of the ones that you mentioned, the cucumber mosaic virus is one of them. And I don't remember, there were a couple of others.
Mosaic, I think, was another one.
I think that might have actually been one of them as well. And potato virus Y or X, one of those two, and a couple of others. But yeah, so basically, I just want to say that as an integrated pest management specialist, it's very important, I think, for people, especially when I'm talking to people about diseases and pests and what they can do. They don't always have to know everything there is, but if you are a cultivator, it does help to know a little bit about how your plant's health actually affects antagonistically things like pests and disease and pathogens and things like this. What is the actual thing that's happening? Because then when people tell you that they have like a product or a service or something that will help you, they can actually know kind of mechanistically how that happens, at least generally. And it does go a long way to like demystifying some things that maybe aren't true or are a little bit more nebulous. I bring this up as a long-winded way of just saying that I'll explain two ways that plants do what you just asked. One is the hypersensitive response, which is also true in mammals and animals. That's like programmed cell death. So to affect viruses, if plants have a really good hypersensitive response to that virus, if they're able to detect particles or things associated with the virus and then mount an immune response that is actually effective, then they're kind of winning that battle really well, because fundamentally that virus requires the cell machinery to work and to replicate itself. So if that plant is it just has a robust immune system for detecting and then kind of quickly priming that response, which happens a little bit different in different plants. But if it has that ability, then I feel like that's going to be something that's really important. And so that would be one thing that might make a plant resistant, for example. And as we actually sort of genomically sequence our plants and test for resistance genes or gene-based resistance in plants and what they do, we'll be able to actually prove that, or at least confirm that one way or the other. The second way that plants do this is through what's called RNA interference, or basically gene silencing. So they're able to kind of either cut or kind of interrupt or disrupt the ability for the genetic material from kind of getting replicated and processed. And you can do this at various levels and at various points in the process. And I'll be honest, I don't always understand all the little intricacies that have to do with that. But those are the kind of the two ways. So it's totally within the realm of possibility that a plant can be resistant, but that's going to be kind of either cultivar specific, sometimes individual specific. And there are other factors besides even genetics that can have this effect or can facilitate it. Like obviously, all of these defense mechanisms require energy. They require, metabolic activity to be good. And, if you're water stressed, because usually in the wild, plants can't move about, so they rely on their ability to deal with and kind of de-escalate the stressors that they deal with. Maybe it's really hot, maybe it's really cold, maybe there's not a whole lot of water. In addition to, I got bacterial pathogens, I've got fungal interactions that are directly good or directly beneficial because they mine me my phosphorus, or they are doing whatever other things. There's this massive array of things going on.
So let's talk a little bit about, I didn't realize there was a difference between hop latent viroid and hop stunt virus. I thought they were the same thing. Do you want to touch on that? That was news to me before I was doing some research for this show.
Oh yeah, and I think we might have actually talked a little bit about that on the Chief Homegrow podcast. It might have been or maybe earlier, sometime we were talking about. I think I remember saying hop stunt. I think I did remember something noting that a little bit, but yeah. So hop stunt virus is different. I actually don't remember the genus off the top of my head. I could look it up right now, actually. At the IS, that same committee about viral taxonomy. Internet. I know, right? The internet. Hop stunt virus, right?
Yeah. Hop stunt virus.
Yeah. Actually, so this is a great example of one of the problems with talking about viruses is that their names get changed a lot, especially over time as we merge them with other genera, or we realize that less so now because we have a lot better ways of analyzing them. But actually, I'm not going to bog you down with the search. It did not take 2 seconds to find out. So maybe other people can look into that too. But yeah, they are different entities, essentially. But I can't go into too much more detail. I was not primed for it. Just like the plants.
I've often heard that it can be some of the hop latent viroid specifically can be transferred through pollen. Has that been confirmed, or is that just suspected? I didn't know.
Oh yeah, that's a good question.
You would know the answer.
I actually think that's not the case, or at the very least, it might be super uncommon. See, right? When it comes to empiricism, it's very hard to prove a negative. So what people end up trying to do is trying to find situations where something isn't true, like through falsifiability as sort of a proxy for it being the case, or they see if they can find it supported. And there's not a whole lot of research on it. So I a little bit feel uncomfortable saying definitively one way or the other, but it's kind of also like with vertical transmission in general, either through pollen or through the ovaries of the plant. Some reports show situations, at least in hops, where it's kind of very mild. And some also research reports are also just kind of old. And I feel like, a reinvigoration of that research is already on its way, and we'll have better information. But at the very least, I think that if we take all of the research that we have currently, I don't think that it's something that's at the very least very common. Because sometimes you do get transmission that's vertical, but it's very inconsistent, or it kind of only happens in laboratory settings to see if it's theoretically possible. But in the field, the chances for it might be incredibly slim. And so that's my take on that subject.
All right, let's talk about everyone's favorite mosaic virus. I see this almost at least one strain in every facility I go to, it feel like lately. What are your thoughts on the different types? We've gone over, there's lots of different ones. I wanted to also bring up, I figured this would be a great time. If you guys ever are curious about your stuff, currently, as far as I'm aware, the company that has the widest range of testing for the different types is Agdia, AGDIA. And they have beet curly top virus, hop latent viroid, hop stunt viroid, alfalfa mosaic, Arabis mosaic, cucumber mosaic, and a whole bunch of others, tobacco streak virus, tobacco mosaic virus. I think they also have tobacco ring spot virus as well on a different pitch. But they have a whole wide range of different things depending on how you want to do it. If you have the equipment to do it, or if you want to buy
click
it.
on the hop stunt, see that's hop stunt viroid. So when I was looking up the ICTV webpage for hop stunt virus, I found hop stunt viroid, which is in the positive viroidy. So that isn't actually, so just like we were saying, that is not a virus, but I have heard hop stunt virus. I've also heard, for those who are familiar with it, sun hemp with 2 N, so SUNNHEMP. And so sun hemp is a different plant totally, for those who are just not aware of that. It's a completely different plant, sun hemp. I don't remember the binomial name myself right now, but in case people had heard that word kind of bandied about.
It's a cover crop. Oh, let me throw it up here. This is the first thing I found here. Sun hemp seed.
There we go. Yeah. Crotalaria uncea, I'm going to say. Something like that. So yeah, it's kind of like corn. I don't think it's exactly the same, but it's like the word for corn. Corn actually doesn't refer to specifically maize or whatever. It actually just means kind of like the grain that people make, or the most generally used grain, essentially. But nowadays, like, that's synonymous with corn. So I just said corn, so it's like it's ingrained, right? You know what I mean? But corn was not always corn, if that makes sense.
I found this company too. This is the other one. I haven't used these guys, but I did find these guys. All Crop Solutions. They have a whole bunch of different ring spot, streak, and some other funkier ones. That's
Even
another
phytoplasma.
Yeah.
Phytoplasma, that's nice.
Yeah. So do you want to touch on phytoplasmids? People don't really talk about those too much, and how much of a danger that could be if it really took off.
Yeah, absolutely. Also, I realized like I often do, I didn't actually answer your question about mosaicism. So I wanted to say that sort of mosaic symptoms are just a kind of symptomology. They're just a kind of symptom that you get in plants. It doesn't mean that you necessarily know what virus you're getting. And in some rare cases, something that looks very similar to mosaicism, like we've talked about this, like variegation, and other weird hormonal growths and things like that can sometimes look like something that is like gnarled and you'd associate. But yeah, you should really get it, like kind of you're saying here, get it checked, get it confirmed, because like the paper that I talked about earlier, or what you're saying here, it kind of shows you is that there's more than one that you could be dealing with, and sometimes even multiple, if you can believe it. Historically, this does happen. So and sometimes the symptoms are very, very dramatically different than either one alone. But for phytoplasmas, yeah, phytoplasmas are a special kind of bacteria. They are all vectored by insects. And recently, for example, for those who are familiar, citrus greening disease is a big problem in Florida and California citriculture, and also in China. It's called Huanglongbing, which means yellow dragon disease. And it was recently found that the phytoplasma, at least in citrus, actually isn't a pathogen at all. It just causes sort of like a kind of, for lack of a better term, like there's a specific term that I'm blanking on, but essentially like almost like an autoimmune disease. It's like its presence causes the plant's immune system to go into overdrive, attacking itself, and in the process kills itself. And what people found was when they applied some compounds like from oak leaves and also from some other compounds that would reduce the oxidative stress in the plant, it actually caused the symptoms to go away. And in some cases, even I think reverse, if I remember correctly. And this is like the first example of it in plant pathogen, or at least plant physiology history, which was really cool. I wish I had I could get the report for those who are interested. But there are other phytoplasmas that affect cannabis. There's a couple of different ones. Phytoplasmas aren't very well understood because they've only been known since like, I think the 60s or 70s, I want to say. And so research on them has been very recent. They're not very culturable because they're always vectored by an insect or they're in a plant host. So it's very hard to study them. But from what we can tell, usually phytoplasmas in cannabis cause what's called witch's brooming, which is when the plant produces a lot of like shoots and leaves and other tissues kind of in a very haphazard way, kind of like a rat's nest almost. And so that's called witch's brooming. And that's a symptom of phytoplasmas. Stunting can also be a symptom as well. And if you're noticing leaf hoppers, those could also be a problem because there are some unknown species of leaf hopper in like Nevada, Arizona, Iran, also I think China potentially, where cannabis has been documented with a few different kinds of these bacterial phytoplasmas. I expect, similar to hop latent viroid, they may be an overlooked pathogen that becomes a much, much more massive problem as cultivation increases and cultivators remain rather unsupported, infrastructurally speaking.
So have you noticed any other vectors? I know that or any other things that you think might be associated. I know leaf hoppers have been. We talked a little bit about that earlier. I'm noticing more and more cucumber beetles in grows, especially this year specifically. And I know that they're known for vectoring certain viruses and other plants. Do you think that that might be an issue going into the future, or is there any other ones that we need to look out for? Especially as growers that maybe people aren't aware of or don't know to look for?
Yeah, so a lot of times viruses and their vectors have a pretty intimate relationship. And that's why the silver leaf whitefly is so kind of fascinating because it's a vector for so many kinds of viruses in various families. But generally speaking, I feel like, you kind of have to find that out. Research has to kind of back that up. Usually you don't get kind of, what's the word I'm looking for? I guess inconsistently that can happen in some cases, but a lot of times it's not, it's like just simply like mechanically moving the virus from one place to another. That's usually not the case. But with something like hop latent viroid that is transmitted in this way, like when you cut into leaf tissue, or even just like having leaves like, brush up against each other, that can be problematic. And in that way, again, you kind of want to get research to back that up, but crazier things have happened. But I will say this, sorry, like generally speaking, most plant viruses are vectored by aphids. Some even speculate at over 50%, possibly 75% of plant viruses are vectored by aphids, or at the very least others kind of related, like whiteflies, for example, like leafhoppers, for example, because they have that stylet, and that stylet can pierce the phloem and even pierce cells in some cases without causing a whole lot of like mechanical damage. So they can really facilitate that genetic transfer. So
do you want to talk about aphids for a minute and touch on on some of the different things that people like to say about how to prevent aphids. I know that a lot of people often say if you get your Brix level above a certain level, that will prevent them altogether. It certainly will help make your plant more resistant and produce more secondary compounds to be more resistant to different things, but it's not going to make them immune by any means. Do you want to touch a little bit on this? Because you had a really cool post the other day on Instagram about some research on culturing and things like that, where they were doing. Yeah, I'll let you tell us about it.
Oh yeah, no, I appreciate it. And I do have a source if you want me to stream from my
Yeah, go ahead. I turned it on.
yeah. So how does that work? So do I can just like share screen?
Yeah, just share screen and then pick the screen you want.
Yeah, here we go. Okay, you can see it.
Yep.
Yeah, so this is just an intro. I haven't finished the video itself, but I do go over some of the examples from various research reports. And yeah, when people mention this, it's sort of surprising. So it's usually in the context of like, can you see this part that I'm waving around? Or no, right? Or can you? I don't know. I always wonder if that's actually possible. Anyways,
Yes,
it's here.
We can see your mouse.
Okay, cool. So right. So Brix level, Brix is the concentration of dissolved solids, and in particular sucrose is what we're dealing with here because it's the most common and abundant plant sugar. And Brix is a measurement. Degrees Brix is a measurement of concentration by mass. So we're dealing with something called moles, which is sort of a chemistry term that some people might not be familiar with. But essentially, like 1 mole of sugar is like 342 grams of sugar. It's a lot of sugar, the sucrose in particular. And like when people do research on aphid growth and their ability to feed on plants, a lot of times they have been fed artificial diets that aren't even like from plants. Like they're literally just like, in various cases, as the technology has gotten better, basically they're just large sort of pouches of sugar water. And usually this is in a molar concentration, which is usually like some level of like liter. So like in a SI unit. So 1 liter is about 1,000 grams. So like 1 mole of sugar is like 342 grams of sugar. That means that if you put 1 mole of sugar in 1,000 grams of water, a liter, that's like 34 Brix of sugar. So, you move the decimal point and here you have like 34%. So it's a 34% sugar concentration by mass. And here we have, right, so like this diagram is where I make this like kind of simple equation, because what we see here is dietary sucrose concentration down here in moles. So like these aphids, in this case the pea aphid, it's feeding totally fine on molar concentrations of 1, which is like 34% sugar by weight or by mass. What this actual graph shows, and I can go into greater detail, but essentially as the sugar content ramps up, aphid feeding does kind of taper off, at least like feeding per like feeding session. Essentially they still go back and eat more, right? They don't like stop completely. And as you can see, they actually still continue to drink it up. But this is because sugar is actually it's not actually a phagorepellent, it's a phagostimulant. Aphids love sugar and they process practically all of it. They don't always use all of the sugar that they process. However, they void a lot of the sugar, but it does get processed in their gut. And we're of course just talking about aphids here. I've heard people say that aphids will like kind of like rot on the stem, or they'll shrivel up because of this like osmotic pressure, or that it'll rot. But again, like that doesn't make sense to me. And in fact, this report goes out of its way to literally make that point, that it's or these various research reports to make this point, that it literally makes no sense that aphids would be able to feed on sorry, I've forgotten where I put this actually, but yeah, here. So the osmotic pressure of phloem sap is 2 to 4 times greater than that of aphid body fluids. So an aphid would be expected to lose water to the lumen of the alimentary tract, the digestive tract, and literally shrivel as they feed. Osmoregulatory collapse is circumvented by downregulation of the osmotic pressure of the gut contents, such that the aphid honeydew and hemolymph, which is their blood, basically is isosmotic. So, right. So like, I don't get where people are getting this information from. This is actually by Angela Douglas, Dr. Angela Douglas, who is a plant or insect physiologist, and she's really talented, and she's done a lot of research on aphid feeding in exactly the capacity to understand how it is that insects, herbivorous insects, feed on plants, and how to like use that information in a pest management situation. So for anyone who's been told that like aphids don't have the enzymes or whatever, just putting it out here, this is actually not the case. Specifically, 2 enzymes are responsible for aphid digestion of high amounts of sugar, way more than you would ever really see, or not totally impossible amounts, especially with fruit, but in like the phloem sieve elements, the Brix levels typically don't get this high. Sucrase is one enzyme, and this transglycosidase is the other main enzyme, and these break down the sucrose and also break down the oligosaccharides after that and make it weight, and then that brings the osmotic pressure down for the aphid, and it survives. And yeah, I just want to leave this quote here on that topic. It says, dietary sugars are important not only as the principal source of carbon, but also because they are the single most important nutritional determinant of aphid feeding rate. I feel like that's a pretty solid statement to leave on. So I don't like to take research as dogma. There's always coconuts, no butts, no coconuts, right? There's always coconuts. There's always something that could come up that could make this not true. But it seems like there's massive support for the idea that herbivorous insects can handle high Brix plants and even are attracted to the higher, healthier plants. And intuitively, this makes sense to me, but I try not to rely on simply intuition.
Yeah, I'm definitely seeing a lot more aphids in the last couple of years, and then people waiting way too long to really do something about it. Like if you don't act quickly and you find aphids in your facility, they can rip through a facility very quickly, and people don't really understand how fast they can replicate.
Yeah, I agree with that. Aphids are born pregnant, and even the ones that they're pregnant with are also pregnant. So they're grandmothers before they're born. Yeah.
Real quick, I just want to give a shout out to Fumi's chopsticks. Those are epic, man. I'm gonna have to get me some bud chopsticks now.
Thank you, man. Got to do it.
I think
Then the side effect is you'll probably be better like with chopsticks at the restaurant. People will be super impressed. You'll be like juggling peas and stuff. Like how? Because you learned, getting high, man. You may not be able to tell them that you learned getting high, but
I think of everybody I know. I think Chris Trump has the best chopsticks. He has LED ones that light up, and you can actually like lights up what you're trying to eat, so you can like eat in the dark. I was like, yo, that is the dopest shit I've ever seen.
Has he thought about just eating by a lamp?
I'm not sure. Matthew, what other diseases have you seen a lot of this year? I know I've seen this year's been particularly bad in Oklahoma for Septoria for people that aren't usually
Yes.
dealing with it. What are maybe you want to talk about Septoria for a minute and yeah.
When you said asked me this question, immediately Septoria popped into my head. Also Pseudomonas, both of which kind of look similar, but they are not the same. So Septoria is a fungus and Pseudomonas is a bacteria. And Pseudomonas leaf spot is the disease that certain Pseudomonas species cause in cannabis. And basically, they look like sort of blotches, and they usually associated with like a violet, kind of a violet halo around, I think. I'm not sure if you can throw up. Yeah, here we go. But like Septoria also, so you can see how they both kind of look like this. They have this yeah, like they're sort of the interior has been like necrotized, and then the outside you have this sort of discoloration. And it does mystify a lot of people when they come across it. And I think the thing that number one gets mistaken for these various leaf spot pathogens in general is like a spray burn. Even for myself, like I have to always ask, like, did you spray something new recently? And sometimes that is the case, and the damage can look very similar. So it's always helpful to like consider that potentiality. You can't always tell from a picture or video. It's sort of basic to say that, but it's always helpful to remember and remind yourself of, because even somebody like myself who is inundated with that all the time, constantly in a professional capacity, sometimes your biases do sneak up on you, and you have to remember and abstain from that kind of hasty identification. As for dealing with it, generally, I like to use wettable sulfur when possible. I know that's not always cogent with different kinds of systems, and I think that's totally valid too. But for a lot of people who are growing at home, I do find that that's really great for a lot of different fungi. But of course, sulfur is sort of broad spectrum in that way. So if you have beneficial fungi, it might be a little bit deleterious to them. It might not cause like a sort of like a local extinction event in your grow, depending on how much you use or whatever like that. I also find that clients that deal with this do a lot better if they do cut off the leaves that are affected by the fungus. I think that helps with the reinoculation from happening.
Oh, is there any others that you're seeing aside from those two becoming more prevalent? I know there's quite a bit of things new to learn on the East Coast. I know I'm running into this orange thrip species in Georgia that I have never seen or observed before under the microscope, and a lot more fungal mites that seem to be not really doing any damage to the plant, but just a lot more of them in the soil that seem to be, just stuff that having to familiarize myself with new insects. Have you run into anything new or anything that you think people need to be on the lookout for?
Yeah, I've seen a lot of people ask me recently about Fusarium, which is also a fungal pathogen. And while not all species are vascular colonizers, a lot of them do colonize the especially the xylem water channels, and so it kind of chokes out the plant. And sometimes you see fusarial symptoms as like sort of almost like botrytis for people who've dealt with that in the flower, sort of like a grayish mass, especially on the stem that kind of like is surrounded by like white necrotized kind of like dry plant tissue. That's usually a pretty telltale sign. And especially if you have it on the base trunk, I would expect that plant to be dying very soon after that, especially at that advanced level. Sometimes it causes wilting. And I know a lot of people who have come across, they had no problems, and then like 48 hours passes or 24 hours passes, and suddenly like 6 plants are like wilting, and they're thinking, oh, it's just a water thing, but it's actually a fusarium problem when they go to look at it. And I recently posted about termites. Formosan termites, probably from India, but they're also in various other parts of the world, North America in particular. And they will go after living plants. They'll also go after building materials too and other woody substances, but they will come and they will strip the cambium off of your plants. And the thing about these termites is that they are very difficult to deal with, even when you're using noxious chemicals and things like that. This actually does look like a Helicoverpa larva. So one of your budworm moth larvae, one of the various ones. And that, yeah, you can see the sort of like grayish, moldy, not moldy, but like some sort of like grayish necrotized wounding. And that can often come and become like mold. In fact, I think I might even see in the center what might be like a tuft of gray. Yeah, that could just very well be it. That was a good example.
Yeah, I like that one because you can see how like the little budworm causes the fungal infection above and where he fed, and then his poop kind of caused the secondary infections. I've been getting a couple of decent pictures lately. I had a good picture the other day of a parasitic wasp laying eggs and the butterfly eggs on a cannabis leaf, which I thought was a pretty good picture.
That sounds like a really great picture.
I can pull it up. Hold on a second. What are any other insects? What about borers? A lot of people don't really find those till late in the season either.
Yeah, especially like right around now, or a little bit beforehand, a lot of people were inundated. I was actually going to point out that you brought up a really great sort of unfortunate reality about cannabis cultivation, especially when it's unprotected from the environment and elements. I'm a fan of growing it in this capacity. Oh yeah, that's a nice web of spider mites. But whether it's a budworm moth or it's a hemp borer, Eurasian hemp borer, whether it's the latter bores into the stem and then sort of eats up through the stem and might terminate into the bud, eat some of the flower, eat some of the seeds if you're growing for seed, but it'll generally screw up. Okay, nice.
Yeah, I have no idea what either species is, but I got a bunch of pictures. And then when I switched it to video to try and get a good little video, he took off. And I was like, damn it.
Of course. I know, right? Well, Hymenoptera, ants, bees, and wasps are rife with species that look very similar to each other or are morphologically identical, which could be very well true here. But I want to say that it might be in the Calcidae family, maybe. Sometimes you look at enough wasps of certain families and they just kind of have like what the Germans call a Bauplan, or like a body plan or a body morphology that just kind of like what an alligator looks like, what a crocodile looks like. And so if I said a crocodilian or something, you could like kind of visually kind of think of the physical qualities that they tend to have, and you could kind of know what I'm saying. Or if I say like a reptilian or something like this. So I feel like that might be the group, but who knows? That's a massive family.
This is the cucumber beetles that we've been seeing heavily in Oklahoma this year.
Spotted cucumber beetle and the striped cucumber beetle are super common. And you also see that is the damage that they cause. They are, I think I actually mischaracterized them as flea beetles when they're actually just leaf beetles, and flea beetles are a kind of leaf beetle, but these are not those leaf beetles. They do eat into the leaves, and they cause this sort of like sort of skeletonization or like massive amount of holes in the foliage. And the larval forms, they eat at the roots too, so that's another thing to consider.
Here's another good example of their damage. They'll chew like halfway through the leaf without punching all the way through sometimes too.
I highly recommend the bug salt guns. And you can give them to your kid and just let them run through and you shoot everyone you can find. It's a good time for everyone. I'm just saying.
I own me a bug assault gun. I got one of the first ones when they came out, and I bought very not recently anymore, but I bought their like version 3.0 that came out, and I got the laser sight because I'm a marksman and I want to use that skill to take down some flies or whatever. If I'm
Just got to make sure he doesn't get too crazy. He wants to like, dive over the media bed and like take one out. Got to keep him under control.
grenade out. Yeah. Also, this I have learned from personal experience that the salt will punch through leaf material. So you got to be correctly judicious.
You got to be tactical. That's all.
You got to be tactical. Yes. No, that's an excellent picture, Stephen. Yeah.
I was trying to throw some more examples. I got a lot of them this year.
Right. Unfortunately, right. That's how I feel when people send me pictures and footage, and I'm like, can you get more? Because it would be really great to get more to share with people. And then I always feel bad because I hope they never have pictures and video for me. Sort of.
What is there what about spotter lantern bugs or any of these other ones that people are worried about? Do they go after cannabis? I've heard that they go after cannabis as well.
They do indeed. I have a video on that as well. Spotted lanternfly. There's actually some old Chinese agricultural literature, I think kind of around the Cultural Revolution, that talked about spotted lanternfly nymphs in particular on cultivated hemp. And although there's not been a whole lot of research on that since then for reasons, I wouldn't expect that behavior to change. Plus, the spotted lanternfly, it eats tons of different kinds of plants. That's why we're so afraid of it heading to California, because apparently people have done some pretty extensive and sophisticated modeling of the spotted lanternfly, like environmental tolerances. And it turns out that it would be really, really good in exactly the places where we cultivate orchards and viticulture for grapes, which would be very, very damaging, probably many dozens of millions, possibly even several billions of dollars in damage, just like has been the case in the East Coast. If we have East Coast audience members here, which I'm sure that we do, they will probably be able to tell better than me about how prevalent they are. And they do come from Central to Eastern Asia. A lot of really competent pests come from like the Eurasian area. I think some people even ask the question as to whether or not that's just coincidence or whatever. But I think also part of it's because a lot of plants that we grow for food do come from these areas as well, or at least they have expanded to these areas in the past. And so a lot of herbivorous insects have a predilection for it.
Is there any other good native predators that you want to mention? I'm a big fan of wheel bugs. Just don't pick them up or you'll regret it. What are some other maybe native bugs that people haven't heard of or haven't seen before that they might want to, not worry about if they see them in the garden?
Yes, a big one I like to talk about. Two big ones that people always ask me about are springtails, which I would call neutral to beneficial in the soil. So people get, for very understandable reasons, people see them in the soil, having perhaps never gardened before, and they get very anxious and they're afraid that their plants are going to be eaten. And I've had people take away, they've been like, I can never get rid of these. I've done so many things and I've trashed all of my crops and I'm never growing again. I've had people literally quit growing because of these springtails, which I think is very unfortunate. But they are not a problem in pretty much all cases. I will say that the only case where I find that they are an issue is if they get to be a really large population and you have seedlings and supple tissue. This is also true for my second organism that's not really a pest that people shouldn't worry about, and that's like the mold mites you were talking about. There's various species, but they tend to have like some like long hairs that protrude from their body. People who keep terrariums for like reptiles might be familiar with them. People who have had kind of like vivariums in general, I've even seen them associated. I'm sure you've seen them in like when you grow fish in like fish tanks and things like that. They'll eat all the detritus and the debris and all that free organic matter, and they love moisture. So if you have like a mulch, it can be sometimes very difficult in like a living soil situation to like kind of get rid of them.
Is it the Triphagus mites? Is that one of the groups of them with the fungal feeders?
Yeah, Tyrophagus.
Tyrophagus.
Yeah. That's a genus that's super common. And like some people are allergic, right? So that's another reason to kind of keep them in control. And some people maybe will develop sensitization. I think I've said either on this video or a few others, but like whenever this topic of like allergies and things come up about the mold mites, I like to remind people that a lot of people who drink instant coffee develop sensitizations to a cockroach exposure, and that's because it's in the instant coffee. Not all instant coffee. I don't even like, I want to backpedal a little bit in case people are super grossed out, but it does happen. Bug parts and things do get into processed foods sometimes, and also for that matter organic foods. And some people just have a very low tolerance for that. Yep. So those are the long hairs I was talking about for like mold mites. And you can see pretty clearly the idiosoma is the big body, and the gnathosoma is that like mouth head part. Yeah. And then they have 8 legs, and that's how you know.
Looks
But
almost miniature hairy dog tick.
yeah, kinda. But I think you were also asking other pests that people have been seeing, wasn't it? Or
yeah, pests or beneficials that are native.
yeah.
Otherwise.
Oh yeah, it was beneficials. I wanted to say that hoverflies are one that I love to talk about, right? They the Syrphidae, their larvae, there's a ton of different kinds out there, and many of them, the larvae feed on aphids. And I've definitely seen situations where people have misidentified them as caterpillars, and knowing how bad budworms are, they kill them. But if you do find kind of a weird looking caterpillar in your grow, especially if it's around aphids, then be careful and take a really good look at it and see if you can't find a match with a hoverfly larvae. I do keep some observational footage, many good close-up examples on my YouTube channel for just that kind of occasion. A lot of times they're like a green color, but sometimes they can be like a mottled brown. They're kind of pretty. I feel like the patterning on their bodies is attractive patterning. And they you can tell them different from caterpillars because they don't got a head capsule. Caterpillars tend to have this like hard head with like the eyes, and the big mouth, and hoverflies lack all of that as larvae. They just kind of taper at one end, but they do kind of undulate when they move a little bit. They'll like cast around, and then they'll like pick a place. So on the right hand side you have the head, which is like sunken into the body at this point because it's reached out and hooked an aphid and then pulled that aphid into its mouth. And then at the other end is the kind of like the foot, so it moves in that way. And then I think that was a good picture of a hoverfly that you had on the next. Yeah, a lot of hoverflies, not all, but a lot of hoverflies do tend to have this like wasp or bee-like look to them. This like yellow-black coloration is super common. Sometimes they're even iridescent, like a lot of wasps are. But But as flies, they only have 2 pairs of wings, and they have those little, I'm forgetting the technical term, but those like small antennae that they have. And they have those big eyes, and they also hover in place, kind of like a hummingbird can do. And that's how you know you're dealing with them. And to attract them, a lot of people ask how they can do this. They really like nectar and pollen from like a sweet alyssum, or maybe like some pepper plants, or fennel. I find is really attractive for them. There's a lot of various like herbaceous plants with like little flowers and nectaries, and they love to go around those. Various parasitic wasps will also really love plants like that. So you can get kind of a great population of beneficial wasps and flies that will predate and parasitize aphids and moth eggs and things like that. If you kind of plant these plants around your area, but your mileage will your mileage will vary depending on that area in question.
Yeah, those guys, Aureus, hoverfly, or rove beetles, are all kind of good roaming hordes of murder machines to kill anything that tries to go after your plants.
Agreed. And they all tend to like these sort of like plants with small flowers in like large volumes. And yes, that's what the hoverfly eggs look like this. And so that can be another giveaway, is that these long oblong eggs are around a colony of aphids. If that's the case, you might want to let the caterpillars, you might want to let the larvae do their job.
Yeah, those are definitely a good one to have for sure. What advice do you have for people trying to, and this is one that, even with all the different stuff that I know we still have issues with once in a while. What is your advice this time of year? Everyone's dealing obviously with botrytis and caterpillars. What is your main, what advice do you have for people having caterpillar issues?
A research report actually just came out by a lot of people who are doing sort of hemp research and cannabis research, and specifically they were writing this research about budworms and other pests too, but specifically kind of like the corn earworm that people are dealing with, and the resulting botrytis that it causes. And so the main problem with it is that even if you kill it, because when the moths come in and they lay their eggs on the plant, you've kind of already lost somewhat. You can apply things regularly and sprays and stuff like that, but if you don't catch it early enough, and if you don't have the labor or the time, especially as a home grower, but also at a commercial setting, if you don't have the ability to like have a very vigilant scouting and application setup, which is also costly, then it's very easy for that to spiral out of control. So my main advice to people is that the problem is that most of these caterpillars will be a problem even if you kill them, because they'll either die in the bud or on the bud, and that can cause problems, and mold can grow on the dead body. So that's an issue. So my advice is to eliminate that potential from happening if possible with some sort of physical barrier. And I feel like a broken record because that's what I've been telling a lot of people in a lot of cases. Oh, that's a nice bee fly. Flies don't get their credence for being amazing pollinators. I think flies a lot of fly species do rival various like bees and wasps as pollinators. So not all flies are gross house flies or whatever. So anyways, mesh screen, big deal. Specifically what's called thrips screen is really nice because it also also prevents the ingress of like thrips, even, which are incredibly tiny elongated insects. Spider mites as well, and of course the moths can't get through it, but moths also can't get through like larger pore screen, which might be less expensive or easier for you to come across if you're in a situation where you don't have a lot of options. That's something that you can do too. Mosquito netting would also work if you're growing on a budget and your access is limited. Those are also fantastic choices, more narrow in their scope of things they can prevent. And of course, you have to apply it correctly too. But I find that is the best investment for the money, because you only have to apply it once. And once you've done it appropriately and correctly, they can't get through. It's physically impossible. And the caterpillars won't move from another plant away from you onto your plants. Some caterpillars do this. They have that behavior like cutworms or armyworms do, which are also a problem in cannabis to a degree. But these caterpillars don't do that. So that's my advice for that. Oh, actually, you know what? I do have one other thing about those borers. Now, that's nice. Nice action shot there. That is a robber fly, part of the Acylidae, and it's taking down a grasshopper or some sort of like orthopteran.
Both of them were about 3 or 4 inches long, and they were huge.
I believe it. Yeah, robber flies, perhaps like rivaling dragonflies. I guess they're different weight classes. I think robber flies are like the most lethal and most impressive aerial hunters of their like size category. And that's how they do it. They'll just like literally, they'll take hornets, they'll take grasshoppers, they'll take bees, they'll take anything that's like sufficiently sized, and they'll just bear it down. Doesn't matter.
For those of you in the South, you guys call those skeeter eaters.
Yeah. I bet they take a lot of like mosquitoes and crane flies and things are like no contest. These guys are deft aerial predators. Oh, but I was going to say that for the caterpillars, I've lost my train of thought. I guess I'll also say that like you can get trichogramma wasps for the eggs. They'll parasitize the eggs. So that looks like an armyworm for sure. It could be a yellow striped. I know it doesn't look yellow here, but it kind of reminds me of a fall armyworm or yellow striped armyworm. A lot of the caterpillars of different species look very similar, and they also could have different forms. It does look a lot more yellower when you zoom in. Thanks for doing that. I feel like faintly it's a little bit washed out from the bright sun. Yeah. So that'd be like an armyworm, and those things can crawl on the ground, and they're called cutworms because if you have like a young stem of an herbaceous plant, they'll girdle it and they'll cut right through it, and that is very frustrating. And right now in the Midwest, they're a massive, massive problem. I follow a lot of pathologists and USDA researchers and entomologists on Twitter, and it's a lot of people are talking about it. It's really bad, like in Kentucky and Oklahoma and other places like that. Really wish I remembered what that thing was about the moths. I'm sure I'll remember after the podcast.
This is one of the other super common ones that we see here.
So I know it's a little bit blurry, but it kind of reminds me of some of the tortricid moth larvae that I come across. This sort of like very small diamondback moths are what I think of, which I'm sure a lot of people have actually encountered, especially if you grow brassica plants like broccoli and and that kind of thing, and also crucifers. They love those kinds of plants. And what tortricid moths do is they're called leaf rollers, and so they use the silk and they'll like bind the leaf or the flower material or whatever, and they'll fold it and they'll eat in this shelter. So even if you apply like a biocontrol agent, like a fungus or a bacteria or something, they might not make contact with it, which makes them very challenging pests to deal with.
So this is the one I have no idea what it is, but I've got a couple pupa and we hatched them. So this is the one that's going after the stuff heavily in Georgia. Again, no idea what it is, but you know, if it helps somebody online by showing you this in the adult form, this is the caterpillar that's hitting the hemp out there hard.
I really like that. I appreciate you doing that, because that's all it needs. I actually don't recognize it. It looks like a lot of it looks like a very similar body shape to other moths that I've seen in the past. And I think if I had a key in front of me, I might be able to make a family level identification maybe. But moths truly are one of the, I mean, they're one of the 4 biggest orders. So there's like thousands of species, but that's great because somebody who's more knowledgeable about them might come along and take a look at this. So thank you for sharing that, honestly. And I think it's a male based off of the antennae.
Yeah.
So
yeah, I was absolutely. Yeah. Actually, why don't you explain that to people?
So the antennae, the reason why I say it's a male is because a lot of moth antennae are kind of like pinnate like this. They have all of these little like extrusions, and that's so that they can pick up the sex pheromones and other sorts of like chemical signals in the air, and then they use that to guide them to their target.
We also had a better example of that also on the same trip. One of my favorite moths in North America, a Luna moth. And you can see here that, again, it's a male, has that same structure, the feathery that collection structure. They are freaking awesome if you've never seen one.
I was just, I think you saw it. I was just posting about the Luna moths and how that little tassel at the bottom. Do you remember? Have you seen the post about the not the moth scales as metamaterials, but that it actually is a decoy. It's a biosonar decoy. So if a bat is echolocating and the echolocations bounce off of this decoy here, then the bat will think that that is a body or something that it should attack. It'll think it's like the mass of the insect when it isn't. And so that's really amazing to me because that combined with the moth scale metamaterial research, really, it's just amazing to me, the level of like sophistication that these structures go through.
And a big old fuzzy teddy bears too.
Oh yeah.
Big old fuzz. Anyways, he was on the windowsill in the morning. I thought that was cool. Always a good omen, you know.
I know, right? Plus, if you crush 2 of the wings together and you add a little bit of a giant's toe or something, you might get a antidote. That's what I've heard.
As a
That's it. That definitely is like a fall armyworm or a yellow striped armyworm. It's definitely Spodoptera. Because of the markings, the black and the yellow line, the black holes, the yellow lines, and also on where the head is, that upside down V is pretty telling. Those are pretty common symptoms or morphological traits.
You know, when you get to do outdoor farms, it's all kinds of fun new things to eat your plants. It's great.
At least I guess you know what it is, right?
Oh yeah. We had some of this was popped up. I thought you'd get a kick out of this, just out of entertainment. Someone found this in the back of a hydro store the other day. That was pretty good. Don't ever use that if someone tries to sell it to you, by the way.
In the
What's
back of the
the,
hydro store or under the counter secretly?
One other thing.
What's the active ingredient of that?
Well, it has like uh what's it called? Begins with an A.
Ivermectin?
Avermectin, or not? Yeah, advermectin. Yeah, that's it.
Yeah, it's an ivermectin. Yeah,
Yeah, it's got avid in it. So
ivermectin. Yeah.
there was something else I was gonna ask you, and I fucking forgot it. Shit, I'll think of it. What other things do you think people should know? I guess currently with what's going on, I know that I've found a product called Velifer, which is a significantly more aggressive Bavaria bassiana strain than anything else I've used before. It seems to work on leaf hoppers. It seems to work on aphids, and it supposedly works on spider mites as well. So who knows if it works on any other mite species? But it seems to be just super aggressive. Have you worked with it at all? And is there any other products maybe people aren't familiar with that you think people should know about?
I think we did. I think that is the case. Yeah, but I don't, off the top of my head, I don't remember the active ingredient in that product. Do you? Is it a
Bavaria bassiana. Yeah.
Bavaria bassiana? Yeah. Okay. There are so many products of Bavaria bassiana. It's hard to keep track for me. People who know me know that I do love me my Bavaria bassiana. It's actually, I was reading a research report on it as a myco-insecticide. And I guess it is apparently the most, or perhaps second to like some bacillus species, like the most researched, like basically insect fungal pathogen, which is pretty cool and very vindicating when I talk about it so much. But yeah, this strain PPRI 5339 is not one that I'm as commonly encountering as like the GHA strain, and there's a few others too. And I'm excited because different strains can have different virulence traits and effects, and as we use it more often, we'll probably be able to produce more virulent fungal strains. A lot of Bavaria bassiana tends to be an endophyte in a lot of plants too. And so it might be exciting to have like a dual pathogen virulence that's really high, as well as maybe modulating some of the immune system responses and effects in plants, which it already does in some cases, which are beneficial for the cultivator and for the plant.
What I found interesting about this particular one, there was a greenhouse I was working on. They also had two types of leaf hoppers because of some leaks they had, and people, anyways, long story short, they had two different species of leaf hoppers in there. 48 hours after application, they were all dead. And that was real impressive to me because they've been pretty bulletproof with just about everything I've thrown at them to date. So that was, to me, is the thing that excites me the most about this particular strain. But the fact that it works on all these different soft-bodieds as well as mites is very unique for about, I don't know of any other strain that's, advertised for two-spotted spider mites. That's very
actually, it's funny you mentioned it like that, because I agree with you. In some cases, I see Bavaria bassiana. I've even seen research that shows mycosis of spider mites by Bavaria bassiana, and I think other fungal pathogens too. So it's definitely documented in literature, but I think the reason why it's often not advertised this way is because the mite physiology is a little bit different, and it's possible that that's one of the big benefits of this strain, is that it might be more optimized.
Sorry, there was a funny meme somebody made about our episode. I'll have to send it to you. Or Fumidor put it in the private chat. It was pretty good. Do you have any new content that you're got coming out soon? I know you have the aphid Brix video and some other cool stuff coming out. What's some of the other cool stuff people can look forward to on your channel and your content?
Yeah, absolutely. So I do have the video about aphid feeding coming out soon, probably in this coming week, although I try not to get people's hopes up. I have, of course, in November, your webinar series that I'm excited to talk about. And we'll be talking more about kind of the things that we talked about here, various pests in cannabis, things to watch out for, new pests, old pests, and also like kind of like some of the things about like Brix and general plant health research, which I think would be really interesting to go about. And cannabis research in general is getting a lot more funding, so I'm very excited to see that. I have other projects in the works, but I cannot talk about them yet, not to sound too ominous, but it will be fun when I do get to talk about them soon. A little bit boring, huh? But I guess that is what's on my mind.
No, so you're regularly on, I'm sorry, it's escaping me. What's the name of the other podcast that you're on every week?
Cheap Home Grow podcast.
Yeah, tell everybody about that, because that's a really awesome resource and you put a lot of time into that every week.
Yeah, so the Cheap Home Grow podcast, right? I've been doing it for about 2 years now with various other people. Jack Greenstock is the current host, and I think we even have some people in the YouTube chat currently. Tao, the American one, is there. We've had Chad, not a regular panelist, but still a friend of the show, as far as I'm concerned. And other people as well that I recognize in chat have also been on the Cheap Home Grow, have either been on or attend the Cheap Home Grow podcast. And we talk about various topics, honestly. Brandon Rust is on the panel. Spartan Grown from Instagram is on the panel. Predicated breeding, which is now pure breeding, I believe. And Noah the Grower, and various other people too. And we're trying to get more people from the chat online. So if you're interested in that, we're definitely taking people on to talk about their grow, talk about their situation, and guide the conversation. And I think that's very, like somebody, I believe Fumidor said earlier, it's very Socratic, that way, ask questions and just simply be flexible, because I want to answer questions relevant to what people are dealing with and not be like didactic and telling people what is all the time.
Very cool.
Oh yeah. So what about you put out a lot of other content on your Patreon and some of the other stuff. What's some of the other cool content ways people can get your content and learn from you?
So I do have a Patreon. I do get a lot of messages on Instagram and other social media platforms for all kinds of things, including even some clients and sort of business venues and things. So that's the hallmark of the future, right? That's how it is now. And I don't knock it, but I do try to help as many people as possible. And so if you do have questions, although I will eventually probably get around to them, I'm quickly becoming aware that is not always going to be the case. I now get dozens of questions per day. So I'll have definitely in the low double-digit messages every day. Sometimes it's continuations of conversations from before, sometimes it's new people. But I'm always happy to answer people's issues, especially when they're super simple. That's why I have the Patreon now. So I check that much more regularly. It's easier for me to check it, and it's also easier for me to give information and link to videos or articles or things like that. It's just a much more conversationally geared system. And my Patreon is at the very least $1 a month. It's just much more important to me to be able to help a bunch of people as possible than it is for me to gouge people for prices or whatever. But I do have various rewards for people at different tier levels. So if you want me to take a stab at a subject that you're really passionate about, or if there are questions that you are more curious about, you can come across them, or they'll be given higher priority if you're at a higher tier level, essentially. And at the highest tier level, You can even have your own video essentially, and whatever topic you'd like, and that rolls over month to month. So I think that's been a really cool way to engage with people and answer relevant questions for folks. Yeah. And of course, professionally speaking, I have zenthanol.com where you can contact me there. But to be honest, and also on Twitter, I'm at SyncAngel, as well as on Instagram at SyncAngel. But yeah, I've gotten a lot more of my clientele from Instagram, interestingly. So yeah, those are the various places where you can come across my work. And I'm excited it's kind of still surreal to me to be helping as many as I can, kind of internationally too. I'm sure you've also interacted with that as well, Steve.
I was going to say you did a presentation for the Chinese Aquaponics Association on insects and stuff, and all kinds of.
That's true, and I really enjoyed the opportunity for that, and I appreciate you facilitating it.
There's a huge need for people like yourself that are, both, helping people on a commercial scale, but also putting out good information, or at least identification information, so people are aware of what the heck is going on. There aren't a lot of good resources that people can have, the level of access that they can get to you. And you're definitely one of the great resources that the community has in terms of pest management and stuff like that. So thanks for all the work that you do. If people find something weird or can catch something or something in their grow, how do you suggest preserving it or otherwise capturing it temporarily before trying to get it to the right place without possibly spreading it and making it worse?
Now that's a good question. Not that the other ones weren't, but that's a really great question. I should make a video about, honestly. So if it's like a bug-like thing, some sort of insect or maybe even mite, there's a few things you can do. I honestly support people trying to get some basic sort of entomological gear, like what we call like an aspirator, for example. Although there are some disadvantages to using things like that, like because you're sucking in air, and so sometimes you got to be a little bit careful with that. And it's for smaller insects and collecting them, like parasitoid wasps and things like that, and aphids. But you can also just put it in like a container. And like the biggest thing to do is you want to first capture it unharmed as much as possible, because insects and mites are delicate and sometimes fast and difficult. So if you can capture it like in a glass container or a vial or something and take it out of your grow space, you're practically already there. Then at that point, it becomes a question of what you want to put it in for preservation and what you want to do with it to process it. Maybe you want to just take some really good focused pictures. I recommend always doing that, because at least you'll have the picture, right? And if it's a good picture and it's kind of closely focused and it's not super blurry, you can still take it to a professional, especially one that's local to your area. If somebody like myself is not available, or many of the other wonderful IPM specialists that exist out there, or extension specialists. In other cases, you might actually want to send the physical organism to somebody like one of these specialists. And in that case, you might want to take ethanol. Higher grade ethanol is better. Usually in my experience, I like to get like in fact, I have a bottle right here. This Coptech is 200 proof pure ethanol. Might be hard to get a hold of in some places, but that's really great. And it'll preserve them very well. And then you can like ship them to somebody, or you can at least keep them for a long period of time. This is great too, having a little a digital microscope. I have basically something like this. I have a rudimentary tier Dynolite that serves a great purpose. Their higher tier equipment is really good, honestly, but it's a little bit too pricey for me. And the other problem is that I would not want to risk it being damaged, which in a greenhouse setting is very possible. So, it's like I bought some super steel knife that I'm really proud of and I really like, and If I really need to use something that's sharp and it doesn't get dull, I can use it. But at the same time, I don't also want to lose it. So you run into that paradox. In video games, I was always the guy who never uses the potions and the consumable items because what if I want it for something else? And so I have that tendency, and a lot of people do, I feel. But Dynalight's a good place to look for some close-up visual assistance.
And
Yeah, I think. Oh, yeah.
Which one do you recommend? There's all different ones. The entry level are not that are kind of pricey, but some of the nicer
Steve, do you
ones,
even have to ask? You get the baller one. You get the one that's encrusted with diamonds and gold and platinum and everything
I
else.
know,
The
right?
most expensive one, the special edition, the 2021
waiting.
special edition one. What the fuck?
Yeah,
I
you got to get
remember-
the Space Jam 2001.
You need that one, otherwise your garden will
collaboration
suffer.
with
I remember
Nike.
BizCon, the double barrel vapes, and they had one that was worth like $500,000. It was like solid gold and diamond encrusted and all this other shit. I can totally see them doing that with the fucking microscopes, having like, you know, bling scopes.
Bling scopes. I think you've I think you seeded the universe with that topic. Now it's definitely going to come to pass.
You're welcome, internet.
Easy scope.
There you go. The Yeezy scope. I want to say that the thing that the qualities that make some of the higher tier ones attractive to me are things like the ability to control like your light intensity. That can be really useful to not have it just like 100% on or off. You know, having some granularity there can be really great. And I'm thinking about investing in getting one that's a little bit more like that. But you can get sort of microscopes that are like this for a lot less money that still kind of do the job. You're also paying kind of for the software and the support. So that's another thing to keep in mind. It's not just the mechanical ability of the microscope.
Awesome. Well, I don't want to keep you the whole night. Is there anything else that you wanted to mention before we cut you loose?
Yeah, I think so. Whenever I talk about something like like the Brix and the plant health topic, I think it's always important to clarify kind of what I mean, because I think it's something that a lot of people are very very curious about and also very passionate about. Plant health, right? Like when you grow plants, that's the main thing that you're going for. So I want to say that, like, here's the thing about the sugar content in plants, right? So sucrose is the photosynthate, right? It is the thing that the plant is making in photosynthesis that powers metabolic processes, right? So if a plant has a lot of those and is sending those down to various sources or sinks from the source, the leaves, into the sink, like the roots or fruits or aphids, when they put their stylet into the plant and they let the pressure overwhelm them into their body, right? These are all sinks for the photosynthate. That's actually really great. And having a lot of that sugar is a good thing, right? It is, it's kind of like, because when you're measuring that Brix level, you are essentially seeing how good the photosynthesis is, essentially. But like, it's also important to know that like Brix levels, the sugar content is constantly in flux. It has to be, right? Because like I just said, it's constantly moving from the sources to the sinks and in various veins and parts of the plant, it's going to be very different. And that's why also aphids and other insects, when they're, if they are the kinds that only feed on sap or xylem and sap, they are, they're going to be using gustatory cues to sort of tell, is this where I want, is this even a plant that I'm wanting to feed on? And so really, like, especially with sunlight, like radiation for the sun and or whatever else light source is like a really important part of Brix level. Sugar content goes down a lot at nighttime because photosynthate is not being produced. If you're a C3 plant, if you're a C4 plant, it is different, I believe. So that's when I think a lot of that is actually happening. And then that's a water saving and efficiency boosting process.
Can you explain the difference with that? Like what the difference is between those two types of plants?
I can. So not like in super detail. I used to know it a lot better off the top of my head, but essentially, so C3 plants are doing that constantly, doing that photosynthesis process constantly. There's a photosystem 1 and photosystem 2. And without getting super technical, basically, the photosynthesis process is actually fundamentally flawed in that sometimes you make rubisco and sometimes you make not what you're trying to make. And there's like, I think this happens about 20% of the time with the interactions, depending on the plant or whatever. So that's one of the inefficiencies about the photosynthetic process with the C3 plant. A C4 plant is like a bromeliad or like pineapple, for example. And other sorts of like plants like this. And what they do is because of the photosynthetic physiology or machinery, they are able to capture a lot of the precursors essentially for the photosynthesis process, and they capture all of that and they lock it away essentially at daytime. And when it's nighttime, they can open up their stomata and have this like the transpiration occur. And so that saves them water inside the plant too, which is a boon. But also it means that because they kind of have it locked away in this sort of metabolic stasis, they can control it better and it happens much more efficiently. So that's like my summary of the difference between those two systems.
The other question I was curious if you could touch on, so there's an interesting white paper about garlic mustard being toxic to local mycorrhizae, and actually like killing off mycorrhizae locally as a way of anti-competition. Is there any other plants that you can think of that would be kind of the opposite or the anti-companion plant? Is there plants that you'd be like, yo, like keep them the fuck up out of your garden? Like cilantro would be one that I can think of that is just a root aphid magnet. Like root aphids just love to hone in on cilantro, and it'll bring them right into your garden. So what are some of the other plants that you can think of where you're like, under no circumstance did you ever put that plant near your cannabis plant?
Yeah, so there are going to be plants that I'm not aware of that may have this effect generally, and also more specifically to cannabis, right? And that also gets to another myth that I think we talked about when you were on, right? Is that on the cheap home grow, is that like people have this perspective that plants are all kind of like nice and they're not competing with each other and it's all a happy hunky-dory forest. And that's not the case. Plants are constantly competing with each other, at the very least for like access to light, right? For the photosynthesis we were just talking about.
The same way that like coral reefs are a war zone. People don't even know that. They're constantly releasing all kinds of toxic compounds, trying to kill their neighbor, trying to smother them so that they can get that sunlight. And plants do the same thing. They call it, it's allopathy in plants, right?
Allopathy. Yeah, or allelopathy. And so I was told that the hulls of sunflower seeds are phytotoxic to certain plants, for example.
And to fungi, explicitly, people tell you if you're doing mycoculture to remove the sunflower seeds for that reason.
Oh, okay. Another example, so like that would be like from like a perspective, like they create some sort of chemical. The classic example is the jugalone or hugalone from black walnuts in their hulls. That is very toxic to other plants, a lot of other plants that don't have a resistance to it. So like these things do happen. And but as far as like anti-companion plants from the perspective of like pest mitigation specifically, lots of plants. So like the viral plants I was talking about, there's not a whole lot you can do about that. Beet curly top virus, infects tons of different plants, and not even creates symptoms in all of them. So you don't even know necessarily that you're even next to plants that can have it. Then meanwhile, some beet leafhopper comes in, sucks it up, goes to your plants, feeds one time, maybe 2 times, and then now you have beet top virus. So, but if you got root aphids, plants that they tend to feed on, they have 2 main kinds of hosts. They have grass hosts in the summer and spring, and they have sort of an arboreal, sort of like tree-like host profile in the autumn and winter. And so rice root aphids, like your prunus species, so like your plums and peaches and apricots and things like that, they go after other plants too, but they also will go after tons of like feral grasses and things like that. That's super common in my experience. And my rice root aphid pest primer video goes over like rowan and rye and barley and wheat and peppers as well. There's a lot of plants that they go after. So if you're aware of them on your property, or if you're trying to grow kind of like a food forest situation, which I'm a huge fan of, you have to consider the fact that some of these pests will feed on multiple other plants, especially the ones that you're trying to use to sustain yourself. So that's a challenge, I think, in certain contexts.
Absolutely. Do you have any last maybe things that people need to, how would you suggest someone document something that they found and they're like, man, I have no fucking idea what the hell this is? People take like one bad photo of a leaf or 2 and send it to both of us, and they're like, what the hell's going on? How would you tell people how to properly document something if they were trying to ask for help properly? I think getting a full plant picture so I can see how much that issue is spread, getting a top leaf, bottom leaf pictures. What are some other things that people could do to help, make it easier for them to get help?
So, yeah, pictures are a great way to go about it. Like you say, kind of getting a full plant picture perspective is really useful, especially if you have like, maybe you don't know what they are, but if it's like a bunch of them, if you're seeing some sort of dysfunction, you should document it because it could be associated, but it might not be. And so discretion and caution are really key when it comes to like diagnosing problems. Like I said, people they'll see a problem, they'll see a springtail, they'll think the springtail caused the problem, and then they never get rid of them because they're hard to get rid of, and then they don't realize that that wasn't even your problem. But getting a picture of the organism is really crucial. Taking the amount of time necessary, if it doesn't like crawl away super quickly or jump away or something like this, if it seems rather stationary and unmoving, at least taking it, putting it on like a table or some sort of like solid surface to like get a nice, clear, crisp photo, multiple photos, and also video footage is great, not just to send to somebody like me, but to other people, like I said before. And you'll always have that, right? Because if it scurries away, or if somebody kills it, or whatever, you still have that control over that little bit of information, and you can always reference it. When I train people for IPM crop scouting techniques, this is something that I adamantly and vehemently profuse, because I know I've worked with a lot of people outside of the cannabis industry who are kind of traditional farmer type folk who maybe are not very comfortable with computers or things like this, but a lot of people in the cannabis industry are relatively young in comparison, and they know how to operate a camera and they know how to upload to YouTube, and that right there is really helpful. Like here in this video, for example, you can see that the rice root aphid, I love to mention this, their species name is Rufi abdominale. That means red abdomen. And as you can see here, they do tend to have kind of like a reddish orange, like latter part of their body. So sometimes, there is a hint in the name if you're willing to learn the Latin, I guess. So yeah, taking pictures, taking samples, taking more samples than you think you need is very helpful, because don't I know it? Sometimes you will only take a couple pictures and you'll think that's fine. And then you'll say, I'll come back later when the lights are on or whatever. And then they're not there anymore. And, that can be tragic. Although if they're not there anymore, that's also a good sign, I suppose.
Just a little bit closer.
Yeah, a little bit. And they can be kind of this greenish shade, and they can also be kind of like a darker blackish color in my experience. So knowing that also a lot of insects can have different phenotypes, especially ones that are very dramatically different than others, can be important to keep in mind. That's why documentation with photos and videos is just as important as writing it down. Having some sort of a journal or some sort of like a spreadsheet or something that you use to track.
Also, this is a nymph. This is the younger one, completely different color. They're orange.
Yeah, see, that's like a yellowish color, right? So
yeah, and then
I've also
the flying, you have the winged adults, which are in that video that I had, and those ones look completely different. Again, their body structure is completely different. So sometimes you even have insects that have lantern bugs, stink bugs, have radically different morphologies depending on the age of the insect.
It's what makes insects so successful. One of the main things, like there's two main groups of insects, really three, but two big ones: the Holometabola and the Hemimetabola, I think, or I'm forgetting the other name. But basically, there are insects that start like with a larval form and a pupa form and an adult form, like a caterpillar chrysalis butterfly kind of a thing, or a maggot pupa fly or whatever. All of the insects that are like that descend from a common ancestor. Then there's the other ones, like crickets and grasshoppers, where the young ones basically just look like the old ones, but they're a little bit smaller and maybe don't have wings. The ones that do the best, the most successful, the four biggest groups of insects are flies, moths, beetles, and wasps, ants, and bees. So those all have like a larva, pupa, adult lifestyle. That radical change between the larva and the adult stage allows the adults and the larva to not compete with each other for food for the most part. And that's the key to their success. Plus, they're very short-lived and they rapidly reproduce, so they can evolve quickly. That's some really good footage.
Yeah, Marty and I both have gotten some really nice footage of various insects that we run into over the years with consulting, and we try to, this is some of the different footage we include in our classes as reference material.
It's really useful.
Actively feeding on a root.
Right, and you can definitely see the stylet there. It makes sense that root aphids would feed on the roots, right? Because the roots are a natural sink for the plant sugars, right? So it's right there where a lot of those, a lot of that amazing photosynthate is being passed into the roots. And then many people in your audience already know that a lot of that sugar goes to feed microorganisms, beneficial, neutral, and otherwise. So that's another reason why a high Brix level is super useful for a plant. It gives it a lot of resources to work with for its immune system, and also for its extended immune system, I guess, in the shape of the microbiome. But many other things want those sugars as well. So
yeah, it's always fun to get to show off those funky. I'm sure you have stuff that makes all of my stuff look like garbage because you see way more bugs than I do. So is that some of the different things that you have on in your resources? What are some of the different things that people, you kind of touched on that earlier, but with the different stuff you have on your Patreon?
So I try to make a lot of my information not sequestered away to only my patrons. It's really important for me to make that information freely accessible, but it's the supporters that help allow that to be the case. I've definitely used some of the funds to buy equipment for camera operation or for software and things like that, and other sort of miscellanea. So it's been really useful. But I do like to make that information widely accessible. Root mealybugs, right? Recently, I was talking about a person from Chile who contacted me with root mealybugs, and then I shared that. I had mentioned that I had documented it in the past, but also was documenting it here. And a lot of other people mentioned that they had also seen it as well in their cannabis, and also it's known in other plants too. And I really liked that because not only did sharing it give me more information, but also other people who weren't aware of it now know about this pest. Mealybugs are less common in my experience in cannabis, but I'd rather know a pest that's not well known than not know it at all. But I'm biased.
We've had this was that one other one that I think they're tent worms or silkworms, or I'm not exactly sure the species, but they make the really big, they'll cocoon a bunch of leaves together to make kind of a shelter. What's the name of that one that does the shelters?
Oh yeah, well, there's a bunch that do that actually.
Okay.
But I had mentioned leaf rollers or tortricid moths, and one of the other larvae you had mentioned was something that reminded me of like the diamondback moth larva. And they'll, yeah, that's the problem with dealing with them though, right? Is that they'll spin these silken shelters with the leaves or sometimes petals. And if you apply even like a non-systemic, botanical based insecticide or a biopesticide that they're meant to feed on, and then, have problems later on, if they're in their shelter, then they don't get exposed to it as easily. So that's for the same reason why borer caterpillars are so difficult to deal with, right? Pretty annoying.
Yeah, especially dealing with something that's living inside the stalk really can be a bitch to deal with for sure. Alrighty, well, thanks so much for coming on. Definitely looking forward to your talk at the conference here in November, and thanks for all the awesome work that you do and putting out information.
I really appreciate it. Also, same to you and everyone else who sends me pictures or videos or posted online. I want to echo the statement as we're signing off here. It seems like that cannabis growers are already aware of this, but there's not a whole lot of support for us, and there's not a lot of support in the general community. And sometimes we have to kind of rely more on each other than other sort of support measures that other crops and other avenues are able to get a lot easier than us. And it's for that reason that it's really important for me to share that information, because just the more people who know it, the better biosecurity we have in general. And we've done this for a long period of time, a lot of people way longer than I have. So if I can help those people and facilitate cultivation in general, then I feel like I'm doing a good job, whether that's privately or professionally.
Absolutely. How do people find you one last time and your content?
Absolutely. So if you'd like to interact professionally, you can find me at Zenthanol Consulting. You can contact me there. But you can also contact me on Instagram for that matter, at SyncAngel. And I'm also on Twitter at SyncAngel. And I'm also, of course, beholden to my patrons on Patreon. So that's patreon.com/Zenthanol. And all of my content, for the most part, my most informational and crispest videos are found on my YouTube channel, which is also called Zenthanol.
Awesome. Well, thanks again for coming on the show. It was certainly awesome to learn all about pest management and viruses and all different types of fun stuff. And Brix and all the other wonderful things that we went over this episode. And I really appreciate it.
Absolutely. I appreciate being on here.
Fumidor, how have you been doing? Anything new with you since last couple days? What's new in your grow?
Doing all right. I don't know, plodding along basically. Like no news is good news a lot of times with a grow, like honestly, no reports, nothing. Like for example, I've struggled with spider mites for a few months basically. I think I've turned the corner with those. I think I finally dominated their little bitch asses. So that's delightful. The plants are I don't know, you can see it, I think, and how do I put this? Maybe not in every garden, but in many gardens you can see when a garden is just vibrant and happy, like it's past a corner or it's turned a corner or whatever, and you can just see like, oh, all the plants are cheerful, happy, their posture is correct, their leaf color is correct. Like, there's probably substantive things to that. But I was going to ask before we take off for the evening, Zenthanol, how do I put this? Like it was such a refrain among a lot of, I think, well-intentioned people that, hey, if you just increase the Brix of your plants, you'll make them invulnerable like Superman. And I feel like maybe we glossed over that too much. I mean, I know that you've talked about it a few times, but I don't know. You can put that basically to bed, right? Like basically, people went so far as to say you could take a healthy plant, a super healthy plant, I guess, with 28, 29, 30 Brix, whatever, so some super high Brix, and you could put it in the middle of a room just absolutely infested with spider mites, and that plant supposedly was going to bounce it off. And I just always thought that was implausible. Like honestly implausible. Like you would maybe be resistant in that circumstance, but you're still going to get spider mites no matter who you are, like unless you were Superman with like bulletproof skin. I mean, that's absurd, right? Like high Brix is not Kryptonian or something, right?
Yeah, that is, you're right. We might have glossed over that small point a bit, but that's kind of why I wanted to belabor it at the end, which is that I'm not saying that high Brix is bad or not even relevant or not important.
Sure.
For the reasons I mentioned. But yeah, it's mostly that statement that like aphids will like rot because they don't have a pancreas or something like this. It just kind of flies in the face of the admittedly sort of unique information, erudite information about bug physiology. Like who has the time for that? I do. So that's why it seemed rather absurd to me. And it also doesn't match up with my experience because I have worked with many people who use Brix counts and they have had pretty high levels of Brix and they still get pests. And so, yeah, I just felt like that was sort of a over egging the pudding a little bit. And I think also it bothers me a bit because not only is that information not the most accurate as I understand it, but also I think people get this idea also that like that means that their plants are inherently unhealthy or that they have somehow failed to cultivate their plants appropriately. I think I've even heard people say that if insects feed on the plants, that means that the food or whatever resulting product from it is like inherently like not food or not healthy, but like not even like suitable for human consumption is the like words that I've heard like said explicitly. And that just doesn't make any ecological sense to me.
No, I mean,
so I mean, I don't have an explanation for it. It just doesn't make sense.
I don't understand that either. I mean, obviously, and we've talked about this before. I mean, a higher Brix plant is probably doing better overall, but it doesn't necessarily mean that it's going to be, magically immune to stuff, which I think is how it's often explained.
It's one of the reasons why I like to focus on like genes and like the molecular interactions that are happening at that like basic level, because it helps illustrate that like arms race between like the plant and the herbivore that's competing with the plant's immune system. And that's why I like to bring other like quirky ecological examples with like viruses that are good for the plant. Curvularia thermotolerance virus. If a specific salt grass doesn't get this virus that's in a specific fungus and that colonizes the plant, it can't live in its natural environment. Or the fact that some aphids, because they do secrete honeydew, which is a cost to the plant, right? They're sucking out the sugar. That honeydew doesn't always get wasted. It can attract ants that help out the aphid, defends the aphid. But the aphid parasite is actually beneficial to the plant because in some cases, those ants will attack and chase off or kill more damaging herbivores. And so from that ecological perspective, although it takes a cost and resources, it actually benefits the plant in aggregate, if that makes sense. And of course, everyone knows examples of things like monarch caterpillars and honeybees and other things that make use of plant resources in one life stage and then benefit the plant in another life stage. So these interactions are complex, and I think they sort of defy simplistic viewpoint.
I remember there was a plant, a flower my grandmother used to grow, and it required ants to open the flowers up. I can't remember the name of it. I'm sure someone in chat will know. But this was one of the more interesting ones. I actually sent this. Anyways, I thought this was one of the more interesting interactions that I've ever read about, and this is about how certain terpenoids are actually change the scent of aphids and caterpillars to attract their host parasitoid wasp. So it's like, imagine a plant inventing a compound that's going to change the smell of a predator that's feeding on the plant in order to attract the parasitoid wasp. Like, these are some of the crazy stuff that we still don't even know even 5% of that's going on in our gardens. I think especially as we learn more and more about the complex nature of cannabis, cannabinoids, and terpenes, and the depth that they have, we're going to discover all different types of crazy interactions that are similar to this.
People mention things like you say, like if going back to the Brix content point, that because the Brix level is high, that means the plant can fuel. So it's a specific fact, there's the specific claim, right, that like the insects cannot handle the sugar, that the sugar itself is insecticidal in some way, or that they'll avoid it altogether. That stuff doesn't make sense. But what does make sense is that it can help fuel immune system responses and the microbiome in the soil and that kind of stuff. And also these sort of ecological intricacies that I mentioned earlier. But when people mention that it fuels these herbivore-induced plant volatiles or green leaf volatiles or whatever, or change the scent of the pest itself, the herbivore, they kind of leave it like at that. But obviously, it's more complicated than that, because that's not a foolproof thing. What if the parasitoids aren't there? What if the parasitoids have become habituated to that scent in the first place? That happens. It's one of the reasons why, like, aromatic profiles and that kind of stuff, we know that they're useful and important when we do like electrophysiology tests and we like try to see, okay, does this odorant get a reaction in this parasitic wasp? Okay, cool. Is that related to this bug in some way? That's helpful. But in reality, these behavioral quirks are plastic and flexible. And as the environment changes, their sensitivities and interactions also change and are in flux. So again, it's kind of more complicated than that. I posted about a unique aphid ant interaction, and I also don't want to keep on going forever, but it really hammered down this point. People who are interested in this kind of stuff should really look into the research of ecologists. In specific, a lot of ecological theory really helps people understand like plant tritrophic interactions between like plant, herbivore, and predator, or plant microbe, herbivore, predator, or something to this effect. There's this sort of contextual webbing. This was on my Instagram. I posted about a report that says, the title is called "A Symbiotic Aphid Selfishly Manipulates Attending Ants Via Dopamine in Honeydew." Basically, not all aphids do this, but this specific species literally dopes its honeydew with dopamine, and it makes the ants aggressive, and it makes them more antagonistic to other organisms. And when these aphids heavily rely on ants, some aphids are a little bit, they can take it or leave it, but these ones really need that. If they don't have ants tending them, they quickly die out in natural environments. And in that paper, they talk about something that's really key about symbiosis that I want to share. And I think at that point I'll be done, which is that Under natural selection, symbiotic relationships are predicted to decay because the cost necessary to maintain the relationship lowers the instantaneous reproductive rate, which essentially means basically if you use resources in the moment immediately, you're obviously going to be less fit in that moment, right? Kind of like getting tired after exercising or something. Good for you, but in that moment, you're weakened, as an example. So it says this cost should invite cheaters that do not pay the cost, but exploit benefits from the symbiotic system. So remember, just because there's symbiosis, there can be things that exploit the same pathways that beneficials do, and parasites often do the same. And so there's two ways that people deal with. I'm not going to read this verbatim, but basically there's two ways: partner choice and sanctions. So partner choice is knowing how to choose a beneficial microbe or something, like using a chemical compound as proxy or something like this, or by punishing organisms that do cheat these systems. So like symbiosis has been around for a very long time, probably since algae and the microbes that became chloroplasts, and the microbes that became mitochondria in animal cells. It's been around for a long time. And so ways of exploiting these systems has been around for billions, at this point, like hundreds of millions of years, billions of years. It's all complicated, basically.
Speaking of it's complicated, I don't know, maybe my last question anyway over the evening, and maybe I'll let you go. And Potent, I don't know what his plans are, but I was going to say, like, I don't know, this is maybe a facetious question on some level, but people always think that there are like magic bullets. They think that it's going to be high Brix or it's going to be silica. People have been talking about like all kinds of fancy silicas that are super duper expensive. Those are going to just change your life in every way. People will love you. Like you'll walk down the street and kids will like laugh at you and your pancakes will be tastier. I'm joking, of course, but I mean, like, is there any magic bullet that you've ever seen, basically? So in other words, in your case, probably that would be bug related or anything else. I mean, whatever. Have you ever seen a magic bullet in growing?
I would say that I have seen things that come kind of close to that. Nothing is perfect. All tools and weapons are contextual. But basically, some things like that are broad spectrum, I think are probably the closest that come to that. Now, some things are broad spectrum and you got to be ecologically conscientious, right? As much as like Beauveria bassiana, for example. And it is found in many other places, so it's not like it's a contaminant. But the strains we use agriculturally are, of course, different than the natural ones. There's real risk that even botanical chemistries we use can affect the natural ecosystem negatively, of course. Even things like wettable sulfur. But like I say that broad spectrum things like the wettable sulfur, Beauveria bassiana, and maybe even some predatory mites like Swerskii cucumeris do come close because they tend to kill a lot of various species and pests. So you kind of get a lot of bang for your buck. But at least in the case of like the sulfur, there is a contextual problem with it, that it is also fungicidal and can have other negative effects on non-target organisms. But I feel like that's the closest you kind of get to it. That and maybe physical barriers that keep the organisms from coming in the first place, and other cultural, what are called cultural controls, right? The how and the what and how to your grow. What are you doing and how are you doing it? And how are you keeping things from infecting it in an IPM context? But yeah, generally speaking, there's no such thing as a free lunch. The oiko or eiko or whatever, the Greek word that means house or resource. It's the word that we use that means eco. It's the same eco for ecology and it's the same eco for economics. There's no free lunch. There's resources being exchanged. Energy isn't being destroyed, it's being transmitted, right? It's being just going from one state to another. So you just have to account for it.
Yeah.
There was a great question from Chad about insect frass and chitinase. Do you want to touch on that? And what are your thoughts on insect frass use in plant growth and how that affects the insects' defenses on the plants?
Yeah. Sometimes when people ask me this question, they confuse two chemical terms that are easy to confuse. Chitinase is an enzyme that breaks down chitin. That ase, like the sucrase I was talking about, tends to mean like it's like an enzyme that breaks something down. For those who aren't aware in the chat, so chitinase breaks down chitin, but chitosan is not chitinase, and there's a lot of chitin and chitin byproducts in frass because chitin is what makes up insect body tissue, especially the exoskeleton. It's a polymer, a natural polymer. So when people apply it, what you're doing to help the plant is that the plant is recognizing, you're basically priming an immune response in the plant because the plant recognizes the chitin, and that's in a lot of plants a signal that says, hey, there's a bug here. I don't like that. I'm going to prime the defenses that will activate and will have negative effects on a chitin having organism. But research has shown that it can be really, in some cases, receptors and sensors in the plant can be very, very specific, and they can be very different between like mites versus insects or different kinds of insects. So it is actually quite complicated in some cases, and research can show what the immune responses is in plants when you have like a pathogen and then an insect and then a mite, or mite then insect then pathogen, or fungal pathogen, and then the layers of defenses that get primed and changed between orders is also very relevant. Yeah, I guess I went a little bit over for that question, but essentially, yeah, chitosan works as like a priming agent, and I think that's a pretty cool thing to make use of. It's also rather cheap.
Oh yeah, I know we were using it in Zimbabwe both as fertilizer, but also mixing it with our IMO collections for trying to collect some of those local predatory fungi that feed on insect corpses and insect exoskeletons, right? So I think you can use it in a lot of different ways for trying to even source and gather stuff that's local, that's not going to have any type of negative impact on your local populations outside of what you're going after.
Chitin's also important for fungal cells. Of course, it makes up fungal cells, so that's another thing that sort of chitin products can help. So there's a priming effect not just for insects but also for fungi, and other context cues are going to guide that immune response a bit.
What are your thoughts on, is there any other things that you think might work for people collecting their own mycorrhizal fungi, or not mycorrhizal fungi, but their own predatory fungi, like we talked about with the IPMO from Korean Natural Farming that Chris Trump was kind enough to share with me, and I got a chance to use a little bit both in Oklahoma and Zimbabwe. Adding the target insects or insect frass or a combination of both as up to 1/3 of your rice for your IMO collections, and then turning that into either IMO2 directly into liquid IMO or an IMO4, and turning that, adding the insect frass and target insects during IMO3, and then turning it into an IMO4 for application. Do you have any thoughts on the future of kind of localized collection of predatory mycorrhizal predatory fungi like local Viroids strains or equivalent strains, or even ones that we've never even documented yet, which I think is probably going to be the case more often than not.
Yeah, I do. I'm super excited about this. I know that in the past, and I'd still agree with this, that like a small critique about IMO is just that you don't always know what you're getting with. You maybe have great reason to suspect, right, because they're growing on that substrate, that they're probably antagonistic. And I think a lot of times that is true. But one of the things that I'm super excited about is that people, you mentioned doing it at home and that sort of resourcefulness. And I'm very excited about people being able to use basically genomic sequencers that are becoming so much more available to people. Shout out to William Padilla Brown. I like his music. I like his style. I like that he is facilitating research regarding things like cordyceps mushrooms and cultivation and that kind of stuff. I expect to see a lot more people like Mr. Padilla Brown. I expect to see a lot more people being excited to find out what it is that is powering the genes that make up these advantages. And if people are able to culture these effectively, and of course you can get contamination and other problems, and it might not be empirical always, but it's like so cool. And I think people will be able to glean more information about those IMO consortiums that they're developing. And I don't know, I think that's going to take it to a higher level than it already is for people. I'm not saying it's necessary or not, but I think that it will make things like facilitating different strains that people can like cultivate, culture, and then maybe even share with each other. I think there's great biosecurity potential for that kind of a thing, in the same way that people have been doing for a very long time with like psychoactive mushrooms, for example, and well, for that matter, other kinds of fungi. So that's something I'm super excited about. It's a good question.
The last question, unless you want to stick around a little bit longer, I wanted to ask you, root microbes. Can you maybe touch on the fact that like root microbe biodiversity really does help translate, or how root diversity and microbes translates into better plant defenses in terms of secondary metabolites? They kind of work like vaccines in a way to stimulate the plant's immune system, or even just exposure to similar type microorganisms can cause that increase in terpenes. And honestly, it really is the only way to dramatically increase terpenes is to stimulate that plant's immune system, because that's how you activate those secondary compounds. Do you want to touch on that and kind of there's so many myths around terps and all the different products that people sell people, and I just would love to kind of dispel some of that.
Absolutely. I don't know everything there is to know about the topic. Bottom line up front, I just want to be honest about that. But yeah, so obviously it goes without saying, but just to say, terpenes are, of course, they're not there for us, they're for the plant. And a lot of times they are some sort of semiochemical or signal compound, right? So they are, they're either going to be, they might be damaging something, they might be negatively affecting something by being toxic, or they might communicate something, they might repel, they might attract, they might do various things. They might do multiple things, in fact, and usually they do. But how do I put this? Like exposure to various, so alpha diversity, I'll put it this way. In ecology, we have a term, it's called alpha diversity. Alpha diversity is basically like the general, like you might consider like the average diversity. How diverse is it? The alpha diversity will kind of tell you that basically you have a lot of different species in the area, they're very different, or you have not so many. And then also talks about the proportion too, but I won't get into that. But essentially, how does that help you? Well, having even things that might even be like somewhat pathogenic, or or at the very least they might have a lifestyle switch, or they're capable of it, because not everything is just going to have one lifestyle. A lot of these microbes have multiple lifestyles that allow them to be really useful, and have various kind of niches in ecology. So when you have great diversity, part of that is also like things that you might not want to have in that area. But when you have this great diversity, essentially it's kind of like having a tight network. You have more netting in your net. You have more connection points. You have more sort of structural support. Have you ever watched the video series or the TV series, The Expanse, the sci-fi show?
Yep.
Anyone? Yeah. Um, do you remember? There's an episode. I don't remember which one, but one of the characters is like a botanist, and he's explaining to their frankly psychopathic gunman character, who I'm a big fan of actually. But he's a kind of a cool guy, very funny. But he was explaining something that he called like simple complexity or something like this, and it's a great scene if you can find it, because what they're explaining is because they're a spacefaring group of people, of humans, right? And they're talking about how they can use a limited level of like plants and air conditioning. Yeah, there it is. That's the guy in the center is who I was talking about. Very, very interesting fellow. But the botanist is the guy on the left, and he's talking to the guy in the center about how a simple complex system is very susceptible to problems, because while it does have some level of layered defense because of the and redundancy, it doesn't have a whole lot. A natural system generally, not always, but tends to have so many other layers built up on other layers. And so you can have a lot more redundancy. Disturbance is a lot less disturbing for that matter. And so if you have diversity, that's very helpful. If you don't, if you have like limited diversity or some sort of like narrow band, then you necessarily have more susceptibility to disruption and things going wrong, and things going wrong very fast, kind of having like a cascade event essentially, if that makes sense. So that's like kind of the sort of ecological reason why you would want great diversity. But as a cultivator, you don't want like, maybe some people would contend this, but you don't want like natural or completely natural, or you don't want to like necessarily allow what I'm trying to get at is that there are pathogens that exist in nature that you don't want to allow in. There are pests that are in nature that you don't want to allow in. So obviously you are not, as a cultivator, you are trying to like exclude some things, but a lot of like things are not something you want to exclude. And that's why I said the IMO techniques would be, I think, benefited a lot by the sort of democratization of analysis technology, because it will allow clever people the tools to maybe even hone and fine tune IMO processes and strains and cultures. And then those become resources that people can use locally, and also possibly even share with other people. And who knows, maybe it'll be something that people can actually start businesses related to. And especially if people are able to prove that the cultures that they have are the cultures that they are, and prove that the like virulence that they purport to have, or some sort of insecticidal effect or plant priming effect, is what it is, by being able to say these genes control for these effects in this plant. Here's the data, here's the research about it, and here's the culture, and here you can have it. And then I think that could have some really great biosecurity implications. But I think that's for the future, maybe in a decade or so.
That's a great point too. It's something that I often cite when people are like, why do you need to have silica in aquaponics? And it's like, well, there's literally like 600 different white papers on silica and the activation of certain genes in plants. If silica is present or not present above a certain level, it won't turn on certain immune system response genes that actually help a lot with mold resistance and fungal resistance and other things in the plant as well. In cannabis, it increases trichome density because it's used in trichome production, and a whole bunch of other things. So it's kind of, almost goofy to me when people leave out some of these different elements that if you look at a chart and see what's bioavailable, silica is the highest, most bioavailable thing in almost any soil system, right? It's the first thing on there. So it really is kind of a goofy thing, but there's so many different papers, if you look at broader agriculture, about specific levels for specific minerals or the presence of specific fungi or bacteria to activate certain genes that can cause X, Y, and Z. And I think that we have almost no research on any of that stuff for cannabis. Very little research do we have on those. And we have such a much more complex output of secondary compounds with cannabis. You have over 200 or 300 different types of terpenes. You have all different cannabinoids, all different flavonoids, the phenols, and everything else. So it really gets, who knows what we're going to end up discovering with all the different combinations and possibilities there are with that.
And we shouldn't forget that, like you said about terpenes and other things being important for pest abatement, herbivore abatement. Pest is a human concept, right? Let's just get that out of the way. There's no natural pest. They're, in ecological terms, they're parasites, or they're mutualists, or they're commensalists, or they're herbivores, perhaps. But yeah, right. Like stressors have, stressors facilitate the production of defensive compounds, many of which are esters or terpenes or things that we want in our product. On the Cheap Home Grow, I love to talk about how grape growers will control the moisture content that they give their plants. They will control the amount of, to some degree, dryness or aridity in the soil, and other sorts of things like this, in order to kind of, for the end product, create a consistent product that you want with certain amounts of sugars and acids in the fruit. Same thing with peppers, right? You can introduce a little bit of damage to the fruit, and that'll make them a little spicier, right? Because what's the capsaicin actually supposed to do is supposed to be antagonistic to mammalian herbivory in particular, and I guess insect too to some degree. But birds, by and large, are not affected by it because they are the carrier system for the seeds, at least ancestrally speaking, right? So letting them get dry, letting them get hot, these stressors initialize responses that the plant, has evolved in this way to do, because ancestrally that helps them survive. So keeping your plant, that's why plant health is such an odd concept, that we should really consider what we really mean when we say plant health, right? Because cultivators and organisms living in nature don't always have the same exact, driving forces behind them, essentially. A plant can get ate up all to hell, have all of its leaves defoliated. Some plants literally do this seasonally and they get by fine. But from a cultivator perspective, you would never want that, right? So obviously, we must make the contention that these are not the same context, right? Like those dynamics are different.
A good example would be like CBL being expressed much heavier in cannabis in the presence of powdery mildew, right? Like what if CBL becomes like the next big anti-whatever drug and helps people with some major disease, and you have people intentionally infecting their grows at like 2 weeks before harvest with powdery mildew for the expression, or some other crazy. I'm not saying that's the case, but you might have something that's equally as wacky and insane sounding currently that might end up being the commercial main production method, 10 years from now.
What if you inoculate your plant with an endophyte, and especially an endophyte that's been cultured and over many generations to have certain effects on the plant? Like maybe you inoculate with this endophyte, and you pretty much are guaranteed a 5 to 10% increase in certain terpenes in certain cultivars. Like that would be amazing. Yep. Plus give it like other effects, maybe like immune system responses and other sort of acclimatizations. I think that's a really cool prospect, and I'm excited to see. But we have to know this sort of like microbe molecule gene interaction. If you don't know that, you can't. It's hard to make you can't statements of confirmation. You can speculate and you can know that this is the case generally in a lot of plants, but you really want to know for sure. Otherwise, you're spending a lot of time and effort on something that's a maybe. And you also want to it's also hard to quantify certain things too. So sometimes at the end of the day, you just won't know. So I'm hoping to see a little bit more demystification in the future.
Yeah, absolutely. And it's super hard to track a lot of that stuff too and figure out exactly what's going on. A lot of those pathways and stuff are just really hard to, directly prove. So short of just, hey, the introduction of this does this, and without it, it does this,
Yeah, absolutely. You never know what your specific situation is. So that's why I think context is so important.
All right. Well, I don't want to keep you all night. I'm sure we could keep going for a long time, but I'm sure you need to get some rest here as well. So I'll let you go. And thanks for being on. Why don't you tell everybody one last time how to find you? I know I said that once already, but we'll do it again.
Of course. Yeah, no problem. And this is how I am. I can't help but talk about this stuff. So you got me again, like another 30, 40 minutes. But that's fine. I think it's generally beneficial. So you can find my content in multiple places. Instagram, Twitter. My handle is @SyncAngel. You can find me for professional inquiries on Instagram, but also on my website, zenthanol.com. And you can also find the bulk of my information about pests and plant health and things related to that in my YouTube channel, Zenthanol, which I normally say is the channel that I am typing on the chat with, but I actually didn't get to do any of that this time around. I did get to steal some glances at the chat, which is why I knew that the Brix topic was so interesting and in demand. But besides that, I really don't know. So I really appreciate the support from everyone, all 67 plus of you watching. I know that number was higher up midway through this, but if you're with us still from the beginning, you're a real trooper.
These are the real loyal crew. Yeah.
Yeah. You're the real symbionts here. You're the real mutualists.
These are the ones we could pilot a submarine, like an aquaponic submarine if we need.
That's right.
Like the world's largest hanging gardens of Babylon, but underwater. If we needed a submarine crew, these would be those people.
These will follow us to hell high water.
At least underwater. Yeah.
Yeah, maybe just underwater.
We can promise fruit.
All right.
I don't know if we can deliver.
What about you, Fumidor? How can people find you?
I have a channel. Wouldn't you know it? I have a channel on YouTube. It's called Fumidor and the Flavors. I have a podcast as well. It's called Chronic Table. You guys can see the name right there, basically, if you're looking at it. If you're listening to podcasting kind of things and you like to, I don't know, do work or whatever and keep your hands busy with other stuff and then not stare in front of a screen, go check out Chronic Table on iTunes, Spotify, stuff like that. Go check out my channel, though. We like to have fun and have fun, interesting conversations. We have fun people like Potent come on and Clackamas Coot and a bunch of other people come on and just shoot the bull for a number of hours. We had quite a long show yesterday. I didn't honestly mean for it to go quite that long, but I don't know, we just had interesting conversations. Charlie's Farm came on and we started to talk about all kinds of history and background of cannabis, and I don't know, we have a good time. So this Saturday will be the next show. We're going to have Brews and Buds, so we're going to be talking about cannabis, but also integrating craft beer and whatever else, tea, coffee. Zenthanol, you're welcome to join us. I know you're a big tea person, so you're welcome to come. I mean, it's brews and buds. We don't specify it has to be alcoholic brews, but I do specify craft brews. So you're welcome to come and talk about tea. And I know, I used to drink tons of Portland's kind of a tea center here, right? Like a tea importing place. There used to be lots and lots of Chinese here, so there's really a deep connection to China and India and a few other places too, but especially tea. A lot of the tea companies come from here. So anyway, I've gotten to try puer teas and all kinds of different fermented teas, and it'd be fun to talk about that kind of stuff on the show. Tea has all kinds of terpenes and flavors and effects and everything else that beer and everything else does too. So anyway, that's the kind of stuff we kind of goof off about, and also joke about random stuff and make fun of your mama and stuff like that. Go ahead, Putin.
I was gonna say there's the monkey pick tea too, and the civet shit coffee.
That's the one. Yeah, they said that's the best. Yeah, you got to pull it out of a civet. So you got to fight the civet first, though. You got to fist fight.
That's right. It makes it worth it.
Climb up to the tree, though. That's the thing. You can't find it. You can't find them on the ground. You got to. The best ones are up in the tree. You got to find the civet up in the tree. You got to insult its mother. Remember what I was saying about Yo Mama? You got to insult the civet's mother. The civet's pissed off, and then you fist fight the civet. And when you defeat it, the civet poops, and then you collect that poop and you
And
drink
you
that
put the
coffee.
civet into the rosin press and you compress
Yeah.
it.
That's really only because
Nothing
your
against-
uncle is Joe Pesci, but that's
Well, you know, what they do is they buy the emu presses for the emu oil.
Oh,
and
right.
You get a whole bunch of civets the same size as an emu.
So is that how they got rid of olive oil from Popeye? Like they just got an olive oil press and
They just put the whole tree in there now. They just stopped caring.
the olive oil. Poor Popeye.
I did want to give everybody and your website. Did you mention your website?
I didn't actually. Fumidoro.com, ladies and gentlemen, go take a look. Genetic preservation kits, I believe they're called in many places. Some people call them beans, but you may call them different names. You never know how people call things, right? You might call your dog a different name than I might. But that was a weird one. Yeah, that was the Bones like, what the fuck? That's whatever. It's probably it just came out of my mouth. What can I tell you? But go take a look, folks. Fumidoro.com, fumesofgold.com also is an easier way to remember it. It redirects to the same place. Go take a look at photographs and, like I say, genetic preservation kits. And anyway,
great photos of the flower that is available there.
Cheers, man. Thank you.
And also shout out to everybody. We are currently ranked top 3% for cannabis podcast in terms of
what? That's incredible.
So
who could possibly be ahead of you? And can we hire somebody to
like Growcast,
like
Irie
them,
Genetics, DGC, a couple of other people that have put a little bit more effort into their podcasts in terms of
time to put the DDoS to work.
Right.
Only room for one. Only room for one. And also to directly respond to you, Fumidor, I would be down for that. I have to find out if that actually fits in my schedule, but I would love to do that. We type out fermented alcohol, tea, or a coffee, or all kinds of stuff. I'm very much
Saturday's pretty low key, you know, like Tuesdays actually. I mean, honestly, now that I think about it, we have been meaning to get some guests and actually like interview them and stuff on Saturdays, more like, you know, microbrew people and you know, beer brewers and stuff like that. But honestly, it's a fun thought. You could come on and talk about tea. Normally we just kind of shoot the bull, honestly, on Saturday, you know, like everybody's welcome to come on, and it's very much like a pub atmosphere, honestly. Tuesday, it's still frankly a pub atmosphere, but at least the first couple hours of the show, I try to have a guest. So for example, this last Tuesday we had Jordan from Growcast, super cool guy, and we got to chat with him. He kind of held court, right? It was still a pub atmosphere, so Potent got to ask questions and Wes Engine got to ask questions, a few others smashed and a few one, but it was still, you know, he was holding court. So it's a different, actually Jordan said that, like, oh, I've never actually been on a show like this. I was like, I hope that's a good thing, because you know, we have a little bit different vibe than a lot of people do. It's not super unique, I guess. It's not super different. You know, it's familiar. A lot of people have seen the Embracing Organics show, and certainly your Cheap Home Grow and so on. It's not super different, but you know, everybody has their little flavor of how they do stuff. So
I feel like, I'll never forget a close friend of mine described Cheap Home Grow's atmosphere as like a lobby, and I think he meant that not disparagingly, but I think he felt like that might be a reason why it was less.
Yeah, you got to clap back. You're like, what? Are you calling me a waiting room or something? Fuck off, man.
I know, right? Yeah, no. It's a little bit less. I guess you could say it's less energy intensive, like you just said, it's low key, and that's I think that's desirable in a lot of cases, and I think that people are really flocking to it a lot, especially now. This was actually a comment that was made like a year and a half ago, so it's a little bit of a different climate now. But yes, I appreciate the invitation.
Right on. Welcome.
And everybody, you can find us over at apmjclass.com if you're looking for a full class. Again, we have a ton of content on there and constantly adding new stuff all the time. And we're also over at apmjnewts.com. And we'll have some cool aquarium kits available there. Sion's working on doing the instructions and the manuals and stuff. We'll do some video content to that here in the next couple of weeks. But we have all your minerals. If you want to buy them by the pound or 4 pounds, or if you want something different, we're happy to sell you that too. Just let us know how much you need. But we'll sell you all the stuff that's in those fancy bottles you have in bulk. I mean, you could spend, you know, $50-$60 on here and make the equivalent of, you know, $4,000 to $5,000 worth of bottled newts and save you a hell of a lot of money rather than buying a bunch of pre-formulated stuff. Or put in your kit, put it in the gallons of your aquarium, how many weeks of flour, and away you go. and we send you some kits that, all pre-done. And see, we've done side by sides with different nutrient kits and everything else. We put a lot of time and effort into developing this stuff. So if you're looking for that type of stuff for turnkey aquaponic solutions, Marty and Roger and I put a lot of effort into making that happen. So definitely check that out. Also check out Marty and his content over at AP Meds, and all the awesome YouTube content he does. And you can check out myself at Growing with Fishes and Potent Ponics on your favorite podcast app. We're on pretty much everything at this point, including YouTube, Amazon, Spotify, all the things. I've put a little bit of effort into getting us into every last thing that we weren't into already the last month or so. So definitely check us out. We've really been growing a lot the last month or two. So thanks everybody for that. And we'll catch you guys again next week. We have... hold on a second. I'll tell you who's going to be on the show next week. I have the guest booked out for quite a while now. We've had a lot of cool people signing up for the show. Next week will be Wendy Kornberg. Oh, no, I'm sorry. Wrong week. Is it the wrong week? No, next week will be Luna Whitcomb, editor and writer for Skunk Magazine.
That's cool too.
Oh, I'm one of those. I should probably mention that.
Right
I'm
on.
one of those.
Well, you can recognize each other to be like the New Yorker people, where they basically like. I
Yes.
guess I should make fun of how, like the New York, the jokes about how the New Yorkers like they meet each other and like huff farts and stuff. So yeah, I'm not going to say that, but there's all
No, I
kinds
haven't
of
heard
jokes
that
about how snobby the
specifically.
New Yorker is. Like Family Guy used to make fun of it, right?
I think,
Anyway,
yeah, I definitely get the like.
YouTube or not, for whatever reason, passionate about this kind of social media, I think is what I read from that comment. What that means is tell your friends. If you guys like this kind of show, if you guys, 58 or 60 people that are watching, if you have friends that grow, honestly, a lot of people have friends that grow. These days, they have folks that they smoke with or whatever else. Just, folks that are trying to get into it or already experts. There's lots of people who might enjoy this content. Or, for example, if I can be shameless for a moment, my show on my channel, or of course, Cheap Home Grow, Zenthanol is on there every single Sunday. So if you guys enjoy that kind of content, literally tell your friends. Nobody else is going to tell them. So basically, like, tell your friends, help us grow, so that basically the shows are bigger, faster, stronger. And honestly, like that, the word of mouth is one of the best ways to spread literally any business. People know that, like anything, roofing or freaking siding or cannabis podcasts, word of mouth. You know what I mean? Anyway, back to you.
Alrighty, and don't forget the conference. You can see that and the stuff behind me. I made a background for it. So November 13th and 14th.